sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
What if you could incorporate next-generation sequencing into your lab and be able to provide genomic profiling results to your customers, regardless of your team's current level of NGS expertise? What if you didn't have to send your samples out for genomic profiling and could minimize the risk of them getting lost or destroyed? What if you didn't have to wait for results? The Genexus system provides a true end-to-end workflow with just 2 user touchpoints, taking you all the way from your specimen to the final annotated report. NGS has never been so easy. You will only have about 20 minutes of hands-on time as you use the 2 instruments that comprise the Genexus system, the Ion Torrent Genexus Purification System and the Ion Torrent Genexus Integrated Sequencer. Both instruments have onboard visual systems to track reagent placement and reduce the potential for manual error. NGS with the Genexus system is as simple and automated as PCR, immunohistochemistry, or other techniques that you are familiar with. And it is just as fast. Results are delivered in as little as 1 day. The solution you've been looking for is finally here. The true end-to-end Genexus system workflow brings fast, automated next-generation sequencing to your lab.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost triple the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have zero DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of Inaba Biotech, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of Inaba Biotech emerged not far from here in Pretoria in the year 2000 while I was doing a research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start a biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aimed to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has Inaba Biotech. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of Inaba Biotech. Let us know your comments, suggestions or questions about Inaba Biotech in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
What if you could incorporate next-generation sequencing into your lab and be able to provide genomic profiling results to your customers, regardless of your team's current level of NGS expertise? What if you didn't have to send your samples out for genomic profiling and could minimize the risk of them getting lost or destroyed? What if you didn't have to wait for results? The Genexus system provides a true end-to-end workflow with just 2 user touchpoints, taking you all the way from your specimen to the final annotated report. NGS has never been so easy. You will only have about 20 minutes of hands-on time as you use the 2 instruments that comprise the Genexus system, the Ion Torrent Genexus Purification System and the Ion Torrent Genexus Integrated Sequencer. Both instruments have onboard visual systems to track reagent placement and reduce the potential for manual error. NGS with the Genexus system is as simple and automated as PCR, immunohistochemistry or other techniques that you are familiar with. And it is just as fast. Results are delivered in as little as 1 day. The solution you've been looking for is finally here. The true end-to-end Genexus system workflow brings fast, automated next-generation sequencing to your lab.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have 0 DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of Inaba Biotech, Africa's genomics company, here from our headquarters in Mackelneck, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of Inaba Biotech emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start a biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aimed to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has Inaba Biotech. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of Inaba Biotech. Let us know your comments, suggestions or questions about Inaba Biotech in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DMB-Seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
What if you could incorporate next-generation sequencing into your lab and be able to provide genomic profiling results to your customers, regardless of your team's current level of NGS expertise? What if you didn't have to send your samples out for genomic profiling and could minimize the risk of them getting lost or destroyed? What if you didn't have to wait for results? The Genexus system provides a true end-to-end workflow with just 2 user touchpoints, taking you all the way from your specimen to the final annotated report. NGS has never been so easy. You will only have about 20 minutes of hands-on time as you use the 2 instruments that comprise the Genexus system, the Ion Torrent Genexus Purification System and the Ion Torrent Genexus Integrated Sequencer. Both instruments have onboard visual systems to track reagent placement and reduce the potential for manual error. NGS with the Genexus system is as simple and automated as PCR, immunohistochemistry or other techniques that you are familiar with. And it is just as fast. Results are delivered in as little as 1 day. The solution you've been looking for is finally here. The true end-to-end Genexus system workflow brings fast, automated next-generation sequencing to your lab.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have 0 DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Imad? Good morning, everyone. Thank you very much for joining us for the second day of this workshop. So we will start with the first session about sample collection and sequencing, and Professor Ganga will be moderating the session. Professor Ganga, please come.
Morning, everyone. Thank you all for joining us today and on the second day of this workshop. I'm honored to moderate this session. I will take the opportunity to introduce myself. I'm Professor Rachid Ganga. I'm at Institut Agronomique et Vétérinaire Hassan II. So I'm a fish nutritionist, so I teach aquaculture. So I hope that you guys had the time to digest all the good information that you were able to gain yesterday. And then you hope that you are ready to go. You have your coffee and for another busy day. So please join me to welcome our first speaker in this session, Dr. Xavier David. Xavier is in charge of participating in the development of applied genomic solutions in the human and food space in emerging markets like Africa, Turkey, Middle East, and Central Asia. Xavier has held previous roles related to artificial intelligence and their applications in breeding industry, especially in cattle, sheep, and goat, through implementation of genomic tools in France and other European countries. So as you can see in the program, this session is more related to industry application. So I would encourage you to ask questions and discover what the industry has in the pipeline to help move forward this research. Okay, so Xavier, please join us. Thank you.
Salam alaikum, bonjour, good evening, good morning, everyone. It's a real pleasure to be here today, face to face. We have had a lot of interaction, I would say virtually, with Thangard, with Wabib, with Apollinaire, and it's real pleasure to be here and to be in contact with all these scientists and as well students that want to implement genomics in Africa. So my talk will be advancing agrigenomics with Illumina technology. So I will go through different topics about us, Illumina, about genomics, our technology at work in Africa, solution and thoughts for non-human application that helps to advance agrigenomics, and I will terminate with the Greater Good initiative to give you some updates. About Illumina. As you know, Illumina, our mission is to improve human health by unlocking the power of the genome. However, human health starts with an adequate and nutritious food supply and a safe and sustainable environment. We all talk about, most of the time, the impact of genomics in oncology, in NIPT, in a lot of different human application. However, one of the key points for us as human beings on planet Earth is to get this food sustainability, food security, and food safety. When you see the diversity of end user Illumina is able to serve, it's quite a wide range of different application. I mentioned a few minutes ago some human applications which have been now a real footprint of genomics, such as NIPT, such as oncology, such as infectious disease. I will come back later with the story with the COVID, but of course with genetic disease and I would say genetic disease and population genetics, our technology help a lot the scientific community to produce some scientific discovery. So, of course, for our topic today, Illumina serve a lot of scientific institute, research institute, working on animal genetics, on plant genetics, and of course on all this industry, the breeding companies, which are I would say using this new technology by implementing genomics in their selection program. Let's see now about genomics. This year, 2023, Illumina 25th birthday. That means Illumina is born at the same time that's the start of genomics. Remember, the first human genome was published in 2003. Before, Illumina started in 1998, producing oligos, then developing bit chip, then by the acquisition of Solexa, developing the SBS, the NGS technology. All the things is together with the development of our company. There has been some new development in terms of potential, in terms of power of the technology, either to discovery or to implementation in a wide range of applications. Two key technological revolution, thanks to Illumina. The first one is for sequencing, the decrease in cost of DNA sequencing. It has been a lot of questions yesterday during the roundtable about what's the final cost per genome, per gigabase produced, and so on. Indeed, when you see, it's a very young technology, less than 2 decades, indeed, compared to PCR, which is more than 4 decades. And you see, it's like in 2007, you have bought an Airbus 380, more than 400 millions of euro, and now you will buy it for $15. So that's really, when you see the decrease of the price per genome, per human genomes, it has been a huge decrease in terms of cost per whole genome sequencing, due to technology improvement in terms of chemistry, in terms of data storage, in terms of data analysis, and so on. Another technology, which is part of the DNA of Illumina, is a microarray, which is a technology when you already select the markers you want to work on. So with a bit chip, you know what you are looking for. And this technology, which is now more than 15 years ago, still is the right answer on the large-scale screening tool. For example, last year, we processed more than 20 million of samples all over the world, or 60% in non-human, 40% in human. What's in non-human? For example, in livestock industry, either cattle, sheep, poultry, and so on. Companies like CMEX, Solexa, all these all breeding companies use as a commodity, this kind of tools. The second application is of course in plant breeding. So you see, these two types of technology is really a tool for you as a scientist in Africa BioGenome Project to know what kind of best technology I can choose depending of the goal. Because thanks a microarray, you can check the diversity within a breed where you already know the reference genomes. Let's have a look on our technology at work in Africa. I joined Illumina 2018, 5 years ago, before I was in the livestock industry, breeding of different types of species, mainly in Europe, when we developed all the data sets and I would say the different logistics to develop this tool in terms of commodity. When I joined Illumina, I had the chance to be part of the Africa Consortium. The collaboration has been started previously in 2016. They developed a content for a chip, the Africa chip, and I had the chance to develop the second version together with Nikki Mulder and her team. Why I'm very proud and happy with this achievement is because it was the first time when a supplier decided to develop a content dedicated for African genetic. We know in human, African genetics is different from the Caucasian one. That means if you use a Caucasian content to predict a polygenic risk course or whatever on the African population, the accuracy of your prediction will not be there. That's why you need your own data set, your own backbone. That's the first point. The second point is that thanks to this project, more than 250 publication has been used using this data. Yesterday, somebody mentioned what's the most important things for you here in Africa to get the sequencer, the last release sequencer in your lab, or to produce data, genomic data. The key point is to produce data. And with this chip, that could be a genotyping by sequencing or whatever, with this, I would say technology, more than 100K samples have been run. That means it is a huge number of data based on the African population that has been produced, which is a foundation for association for a lot of things. That can be an inspiration for African biogenomes once the reference genomes, the new genomes has been discovered, to be able to build up some data set, some association with phenotypes, to be able to use this diversity in terms of livestock and plants for, I would say, the common use or the routine use in the farms. So as a consequence of all this project where we have been involved at Illumina, Africa, but as well all the partnership with CDC for infectious disease like HIV, tuberculosis, we have now a quite reasonable mapping of Africa in terms of capacity. Build capacity, build capability. Two key points. Previously, 2018, I would say South Africa was the main hub, the main, maybe the only one. Now you see, you can have high throughput instrument, either in West Africa, either in Egypt, either in Central Africa, last Nigeria, and you have as well capacity to genotype all over the continent now by using a OCB chip. That's something which is important. There is still some gap. Let's see where they are, how we can fulfill that. But the most important thing to keep the sample first in Africa for the turnaround time, but for as well to help local staff of scientists, of lab technicians to become capable to run samples on the continent. I will not go too deep in our workflow because Mossab from Megaplex will present all our technology. Just something to keep in mind, which is for us important. We will have to deal in Africa Genome with a wide diversity of samples. That means in terms of DNA extractions, you have to find samples by samples, the right technology, the right tool. Then with our technology, we will offer you a smooth end-to-end solutions for library prep until the secondary analysis. Thanks to Dragon, which is on board on premise for some on the Genexus 2000 and NovaSeq, you can have the variant caller pipeline available. That's something which is a really help for the bioinformatician and the bioinformatician as a secondary analysis. So just to conclude this, our technology, the Illumina genomic continuance, this is on one hand, you discover a new variants. On the other hand, if you need to screen a large number of animals to validate some association or to tackle the diversity within a breed, you can use the second technology, which is microarray. Never forget that we have the next step after discovery, after association, that can be the methylation. We will launch next year the first methylation in cattle. That we know that in human, methylation is the next step of knowing the biological path between the genome and the disease, all the performance for plants and animals. That's really something that can be implemented as a reasonable cost on the chip when you just choose the CBG sites over the genomes that are really related with what you want to study. Now, this is a little bit more coming back for advancing agrigenomics. Why is it interesting? Is it important to keep in mind that within the Africa Genome Project, try to keep to discover this, characterize this diversity. What for? To allow the conservation, of course, but as well the food, to guarantee the food security. Two things which are important: food productivity and food sustainability. Two things which are quite important. How to use this diversity, which if you take it as compare to US or whatever other, I would say, environmental agricultural systems, does not seems very productive, very interesting. However, if you take this the strengths of this diversity in terms of disease resistance, in terms of heat tolerance, and so on, and you are able to select it and to keep it within a pool, within a nucleus of animals and plants, and use it by crossing or in pure breed in different types of system, then you can deliver, I would say, a sustainable and productive tool for the local population. Just a few highlights about the huge diversity we have in here. This is in aquatic species. It feed fish, mollusk, crustaceans, seaweeds, and you see only a few of them are already used at the moment used in wild fishes or in aquaculture. Another figures is interesting. We know the top 5 of the crops that produce 80% of the crops, wheat, barley, and everything. That's really something we have to get out of. We know that in Africa, we need some today in Africa, but maybe tomorrow, due to the climate change, we may have need to use this new, or I would say less used varieties of crops. So from how do you advance agrigenomics thanks to Illumina technology? The first point, you have to know the genome. That's the first point. The second point, to tackle the diversity and to see the potential effect of the diversity of specific trait, and then to take informed decision. Know the genomes. This is to get, that's the first, I would say, the first aim of African biogenomes. That means sequencing at high coverage to be able to get these reference genomes, and to tackle as well, to be able to identify very well the different types of species all over the world. Here, a good examples of the yellow fin tuna, which is a first step to resolve the population structures across hair distribution all over the world. Look at in livestock, we have 40 different species in Africa and 8,800 livestock species based on the publication of Hillary. Once we have this genome known, it's important to tackle the diversity within a breed. 10 years ago, the 1,000 genome has been launched in Australia in cattle. And the fact was, the idea was to sequence different individuals from different breeds to be able to see if some causal mutations were I would say, related to particular breeds, and to be able to increase the imputation efficiency. Because once we have the reference genomes, the key points is to have this subset of markers which has in terms of production important, and to use only this one to be able to then to set up some, maybe not genomic section as an ultimate gate, but at least a genomic test. Another examples of the resequencing to tackle these diversities. This is a chickpea association that was very interesting to tackle the diversity inside the species and the association with the right agronomic traits. Another examples, which you have heard certainly, which is a byproduct or deliverables of the Agricultural Greater Good Initiative. 2019, the International Camel Consortium has been the winner of the Greater Good Initiative. There was some quite reference genome and some, but nothing has been, it was single project. In that case, 400 animals representing the diversity of all over the world have been collected in terms of samples and have been sequenced at about on our technology, Nova 6 at 30x. And then thanks to our PacBio Dragon variant caller tool, they were able to have this I would say new information in terms of reference tackles the diversity within this breed. What has been the deliverables in terms of this is the first chip that is now used in Saudi, in UAE, and we hope so in North Africa, to continue the discovery of association between, I would say, a variant and a production in milk for the milk production, or speed for race camels, or meat for the meat camel. Just a remark or a comment, the 400 samples, the challenge here in this project has been the collection of the samples. Not because it was during COVID, of course, COVID did not help, but collecting samples is really something key in terms of logistics. And it's not only the case in Africa, it is challenging in Africa, but not only, as well in other countries. When you set up a kind of project, especially on routine, never underestimate the collection of the samples. Another example is in Australia. It comes from the crop breeding research benefits. A breeding company, it was a cooperative partner with the University of Victoria, decided to sequence as a proper coverage particular varieties of wheat, to identify some which are much more, I would say, associated with heat tolerance. Then, once this marker identified, put on the chip to be able to screen on the routine the different seeds that have this association, this presence of marker, to be able to step by step select more resistance varieties of wheat in Australia against heat. So as I wanted to, and that will be the last point today, agri-genomics is using genomic test as a routine. It's a commodity. You see either in livestock all over the world, US, Australia, and Europe or Oceania or New Zealand. That's really something which is a commodity. Most of the breeding community use it. The question is when this genomic test can become soon a routine in Africa. It will not be the same in the same way. You see, I took other examples, such the Boran cattle in Eastern Africa. Why? Because this zebu, this post-Indica spread, is Eastern African fever resistance. Why I took the endama? The endama is trypanosome resistance. How do we use this natural resistance? Either by selecting as a pure breed nucleus for particular farming system, or how do you use this by crossing with an exotic breed? But we need to keep this nucleus in terms of resource of diversity. Millet is one of the most popular crop in Eastern Africa because it's maybe less productive than wheat, but which is better in the conditions. Yam, cassava, yam 2 years ago, the Greater Good Initiative with Professor Ranjara in ITA. This is typically something that needs to implement tomorrow some very simple genomic tests to improve productivity and keep diversity. And of course, small ruminants, even if it's difficult in terms of collecting data, especially phenotypic data, there is a need to get genetic improvement, as they are really they have a huge impact in the farming system all over Africa. Just to conclude, the Greater Good Initiative, this is a yearly grant Illumina proposed. This year, 443 applications have been received. It's a huge impact. 185 are under review. I don't have the figures of how many from Africa have been are from this 185. So just for information, a lot of them have been identified either fake or out of scope. When I say fake, it's, for example, somebody would just send a CV, for example, or out of scope, the project is about nutrition, about farming system, but not on genomics. So we will have the winner will be announced at the PAG, the Plant Animal Genomics in San Diego in January 2024. And I hope that someone from the continent will be the winner. I thank you for your attention. Yes, no problem.
Okay, thank you, Dr. Xavier David, for your insightful presentation. Just some keynotes. We will have the questions at the end of the session, so please keep your questions for the end of the session. Okay, so our next speaker is Dr. Musab Maloum. He's from Megaplex. He's a business unit representative and a project manager in NGS project manager, supporting laboratories wishing to specialize in NGS by providing them with technical, financial, and scientific expertise. He's trained in microbiology and bioinformatics, and he holds a PhD in microbiology and bioinformatics at the Institute of Hospital University of Marseille. He also holds double master degrees in human pathology and health science from the University of Aix-Marseille and Hassan II University. Dr. Maloum will take us through an overview of what Illumina can bring to our world today across various specialties, with a particular focus on agri-genomics and culturomics. Please welcome Dr. Musab Maloum. Thank you.
Good morning, everyone. Thank you all for being here. Special thanks to Professor Boabid for the organization. Today, we will talk a little bit about Illumina technology, with the title saying Unlocking Genome Sequencing with Illumina. supported by your trusted partner, Megaplex. So we will go through this agenda. We will start by explaining what is DNA sequencing, Illumina sequencing workflow, why you choose Illumina, and a special debrief on Megaplex, your trusted partner. So what is DNA sequencing? DNA sequencing, we just want to read our DNA or RNA. And what exactly we want is to know the position or maybe the order of these 4 bases: adenine, cytosine, thymine, and guanine. That's it. We don't want anything else. We just want to understand what is coming first. It's the A, C, T, or G. And why it's important to know that, a little reminder of the first genetic course. It's because if we have a mutation on your DNA, probably the protein at the end will be altered or modified. So if you have a G, in case of T, you will have a change in your protein. And sometimes it also can cause a termination of protein. And it's simple as that. If you have, for example, TAAA, you will have a brown eyes like mine. But if you have a TAAG, you may have blue eyes like Xavier has. And the trick is to differentiate between A and C and T and G. And this is the aim trick of biotech companies, to distinguish between these 4 bases. Some use recognize the size of the acid nucleic, some use the shapes, some other use the signal, other prefer just pre-labeled fluid. So we need to know that we are starting from far away with Sanger-based electrophoresis. We could sequence half of genome bacteria. It's 1.2 megabase. It's half of the genome of bacteria. And today we are talking about more than 1,500 gigabase a day. And also with NovaSeq X, we are talking about more than 6,000 gigabase of data for sequencing. So the cost of sequencing has dropped dramatically. We were talking about $300 billion for sequencing the first human genome. It took us 10 years. And today, the human genome could be sequenced less than $300. So it's a reduction by 100,000 fold. And not only human, we are sequencing also animals, plants, microbes, DNA, RNA, etc. So what is the Illumina sequencing workflow? It's quite simple. The sample preparation is just the extraction. So there's the first step of the library prep. The extraction can be made by different way: spin column extraction, magnetic extraction, Lee's buffer extraction, or maybe use one of kits of Illumina. We can find the direct FFPE DNA sequencing, the extraction, sorry, or also flex Lee's for blood sample. The second step is the library preparation. I think a lot of you hear this library preparation. What is library preparation? It's simple as you just need to fragmentate your DNA, because we can't sequence a whole DNA. We need to fragment it to be sequenced. And we can choose 2 ways: enzymatic or mechanic fragmentation as covariates. When you fragmentate your DNA, this is library preparation. You just need to put your adapters to your targeted DNA. And we have three different adapters. Each adapter will do his job. So if you see the first adapter, which is RD1SP, it's just a primer that will start the sequencing. The second adapters, the NDX1 and NDX2, will be just barcodes. So barcodes will help us to mix different samples in one run. So this is what we will give us. They allow us to mix different samples, and the barcode will be recognized by the software at the end. And the last adapters is the P5 and the P7. And the P5 have the simple job to bind to your flow cell. So if you never see a flow cell, it looks like this. You have different type of flow cells. It depends on the instrument. And when you zoom into the flow cell, oh, sorry. Okay, the video is not working, but when you zoom into the flow cell, you will find some small oligos that will be hybridized with the P5 or the P7. So when your DNA comes to the flow cell, it will automatically be hybridized with the complementary region on the oligo in the flow cell. So the third step is the sequencing. So sequencing will start by steps called cluster generation. Now imagine you are prepared your library. You have your target DNA, you put your adapters, everything is okay, and then you put it on your flow cell. You need at least a cluster to be clarified, to be clearly detected by the camera. So this cluster generation will allow to the camera to detect this fluorescence, to detect this light from the cluster. And the cluster is just like this. It's one cluster give you a good lighting of your sequencing. And the challenge here is to not have under-clustered library prep or over-clustered library. You need to have a normal one, so you need to respect the protocols. So this cluster generation is made by, I would say, technology called bridge amplification. So bridge amplification is just a PCR. It's a normal PCR of your first DNA that will be amplified to give you one cluster. Clearly, cluster can be detected by the camera. And those clusters are in the flow cell. So the sequencing is also easy steps. You remember we talked about the first adapter, the LD1SP. So this adapter will start the sequencing. And when the primer is hybridized, you will find 4 of your bases, ACTG, and each bases have their own color. When an acid nucleic hybridized to his complementary acid nucleic, it will release a light, fluorescence that will be detected by the camera. And going on, this will give you a beautiful lighting on your flow cell. And imagine the capability of the camera that can differentiate each cluster alone. So this is happening inside the flow cell. So Illumina sequencer are going from small throughput to the big throughput. We have the smallest sequencer, which is the ISEQ one. We have the mini-seq, MySeq, which is the best seller of Illumina, the NextSeq 550, and there's a new generation, which is NextSeq 1000 and 2000, the NovaSeq 6000 especially for Genome Center, and the last NovaSeq X, which is launched in September 2020. Don't forget that Illumina also has a microarray solution that was presented by Xavier, and it works on iSCAN. You can do human genotyping, non-human genotyping, methylation analysis, genome-wide genotyping, target genotyping, and also custom genotyping. So the last thing is the analysis. So you will find you will find three types of software. So there's a software inside your instrument, your sequencer, and this software will give you the image and intensities and the base calls. So it simply take the images and give you the acid nucleic referring to the image. Analysis software, so it can be based on the base space or maybe your own computer, and will give you secondary analysis like alignment and variant detection. And the last software is the visualization software, which give you annotation and reports. So as I say, base space is it's Illumina platform, and inside this base space we'll find some application. Those applications depend on what are you doing as sequencing. For example, if you are doing 16S metagenomic, then we will have an application just for 16S metagenomic, etc. So why choosing Illumina? We have the performance and data quality. We are using a technology named SBS for sequencing by synthesis. And this chemistry, it's highly accurate sequencing. So we will hear a lot about the Q30. We have a high Q30, and Q30 simply means it's a quality score indicating a 0.1% probability that base was called incorrectly. So we are sure that 99.9% that the base called is the base annotated. Second thing, it's compatibility and flexibility. With Illumina, you can do everything. You can do DNA, RNA, methylation, metagenomic. So it's an open system that will enable collaboration and innovation and flexibility. And also, when you are choosing your application, you need to choose the best instrument for the best application. Don't do the mistake that you will choose the instrument and then look for the application. The application are chosen first, and then we look which instrument will be the best for what we are doing. If you want to do some large whole genome sequencing, you can do it on the small instrument. You need to go for bigger instrument, etc. So you can do some small whole genome sequencing on the small instrument like ISEQ 100. But for larger human genome sequencing or plant sequencing, you need bigger machines. And the last thing is our preference of the scientific community. Illumina sequencing is widely represented by the scientific literature. We have more than 30,000 publications on PubMed, and many renowned researcher institutions have chosen Illumina for their genomic research. Great. So let's talk a little bit about Megaplex, your trusted partner. So Megaplex was created in 1992. We are in Casablanca. Our CEO is Mr. Nasser Amrani, and our director is Mohammed Ayush. We operate in public and private sectors. We were the top 334 ranking in 2022 in the biomedical file. And we started, as I said, in 1992 with just hematology. We introduced to the first installation to the public sector in 2003. And we introduced the business unit department in 2020, and we got Illumina in 2021. So we are a big team. We are 85 collaborators. There is different department, market department, commercial, technical engineer, application engineer, etc. We also have a new identity and new challenges. We choose another logo. We got this fresh new logo with unique colors and beautiful DNA inside, showing that Megaplex believe in the new area of future medicine and specialized diagnostics. We are working with different business partners. I think a lot of you know Sysmex, Illumina, Agilent, Grifols, Fluidigm, Generation. So those are big partners that are using also for completing the whole workflow of Illumina. We are working with the hospitals, analysis laboratory, more than 500 private laboratory. We are, I think, in all the CHU of Morocco. We are working with CNRST, UM6P, and a lot of other foreign organizations. We are very present in different scientific days, congress, roundtable. We are doing also webinars. We are present in social network, and we also have editorial content with GBM. And we believe that with scientific and marketing support, we can achieve our goal of capacity building. It's not just about solving problems, but also about fueling creativity, innovation, and motivation for better and more sustainable future for our beautiful country. We have a big technical team. We have 20 field application scientists and field engineer scientists, and 5 on those field application scientists are certified by Illumina. We reached more than 500 training until March 2022. And for NGS, the top application we are working on are agri-genomics with UM6P, LINRA, AGC, infectious diseases with l'Institut Pasteur, INAH, and oncology with private laboratories. And this is our references in Morocco. We have about 13 sequencers in Morocco. So this is where the 13 sequencers are installed. I will finish with a special quotes said by Megaplex team. We are involved in your success because it's also our success that we are measured throughout yours. We count neither our time nor our energy to fulfill our mission by your side. Thank you, and looking forward to meeting you at the hands-on workshop.
Thank you, Musab, for your presentation. So our next speaker is Dr. Islam bin Hassin. She's representing Alliance Global Holding. Dr. bin Hassin holds a PhD from the University of Science in Tunis, where she conducted research at the Laboratory of Molecular and Cellular Hematology in Pasteur Institute. Her research focused on different types of leukemias, such as CML and LAL, and their resistance to chemotherapy. She's currently working as an area sales manager, managing sales for a wide range of scientific products, such as genomics, diagnostics, cell biology, and molecular instrumentation in North Africa and West African French-speaking countries. Welcome.
Good morning. First, I would like to thank you, Professor Balawi, for this opportunity and Ishraq for the efforts. So as introduced, I am Islam. I'm working with EGBL as a area sales manager, covering North Africa regions and French-speaking countries from West Africa. So today I will represent our group as a company, and I hope to tell you more about it. So to expand our company, EGBL is a scientific distribution partner focusing on emerging markets. We focus heavily on expanding our presence within the African markets and Middle East by investing in healthcare and giving access to underserviced markets to innovative technologies. One of our greatest investments is our people. We proudly boast over 35 nationalities within 25 languages within our company. This allows us to always have a collaborative environments. If you look at our history, AGBL is established in 2006 with expansion into Egypt in 2009. From 2010 onwards, AGBL Maghreb, AGBL Africa, Middle East, and Levant were established. From this point, we expanded into Sub-Saharan Africa, West Africa, and the Middle East. Our sister companies were established, Genomics, Abiomics, Avicenna, and completing AGBL and submitting as a global group. To expand a bit more on our various involvements in industry, under our product offerings, we have Avicenna, a specialized brand that focuses on R&D on research and diagnostic reagents. As part of our sister company, Genomics, we offer genetic wellness testing with our Livelands products range and genetic counseling. With our distribution wing, AGBL, we tie our various investments together to offer a comprehensive company serving the customer's needs and the consumer needs. So to expand on Genomics, our main laboratory is headquartered in Dubai, which is the first lab with this accreditation. So it's CAP accredited and ISO certified. We offer high throughput testing through NGS sequencing, microarray technology, and PCR detection. Our unique services cover oncology, reproduction, and rare genetic diseases detection. We complete the services with our, So we complete the services with our products development wing, Avicenna. We offer a dedicated commercial and technical support team with innovative product development in clinical chemistry, molecular diagnostic, and molecular genetics. Moving to our technology offerings, Abiomics supplies bioinformatic tools to simplify data analysis, accessible interface with in-house and white labeling development. It also offers bespoke and customized services. So moving to distribution, Alliance Global remains the region's largest biomedical distribution network with a presence in more than 15 countries. We cover more than 85 markets with innovative products for in vitro diagnosis, life science, and industrial quality control. To start with our portfolio, so we can start with the specialty diagnosis. So AGBL distributes the Cytec spectral flow cytometry, which have revolutionized in the field of flow cytometry. The Cytec analyzer allow for increased flexibility of fluorochrom use, allowing more information to be gleaned from one tube. So various laser combinations are possible depending on the application and budget. And through our partnerships with industry-leading antibody suppliers, AGBL is able to meet your needs for high-quality antibody for all your research needs. So we have antibody for immunohistochemistry, antibody for flow cytometry, Western blot, and more. So also we have next-generation imaging system. The comprehensive portfolio offered by Azure Biosystem means that we have you cover it no matter what you want to image. From the upgradable C-series imaging system that allows you to capture crisp, clean images. From Western blot to bacterial plate imaging, the C-series is easy to use, dynamic system. The powerhouse of imagers, the Sapphire, is the ultimate biomolecular imager for flexibility with customizable and user-changeable laser and filter modules. The Sapphire FL easily adapts to labs' changing needs and advancing research. So we have also next-generation immunoassay, which is the quanteric Sioma technology. This technology enables early detection of diseases that were previously undetectable by currently available technologies. COMA technology has delivered the gold standard and earlier biomarker detection in blood, serum, or plasma, with the ability to quantify proteins that are far lower than the level of quantification. Moving to genomics portfolio, so AGBL is offering NGS ancestors, which are, for example, we have the covaries from the covaries for RNA and DNA sharing, and your gene for NIPT workflow. And for the NGS panels, we are offering for bases and your gene VitroLife. So for bases, it's mainly CIVD kits, which are used validated genetic diagnostic sequencing panels. For bases products are reagent kits for platform-independent new generation DNA sequencing, data analysis software, and diagnostic portfolio. VitroLife kits are for pre-implementation genetic testing throughput embryo, map, and CaroMap kits. And your gene is for NIPT workflow, as I said. So we have also single cell, we are offering also single cell sequencing and multi-omics. Whether you are working with DNA or RNA, your single cell sequencing needs are sorted. So we have the 10x genomics for RNA, which offers 3 platforms, the Chromium, the Visium, and the Xamium for your single cell RNA needs. The Tapestry from Machine Bio is the first and only single cell technology developed to reveal bioinformatics biomarkers that helps stratify patients, senior resistance, and predict relapse. AGBL also is the distributor of PacBio in North Africa. PacBio is providing solutions for both long reads and short read sequencing, starting with the Sequel 2, most recently with the ONSO system and sequencing by binding. Hi-Fi reads or sequencing reads that are both long, around 50 to 20 kilobase currently, and highly accurate, 99.9% and greater, have been demonstrated to have a powerful utility in many areas of genomics, whether it be genome assembly, detection, all variant types, target sequencing, long reads, RNA sequencing, or ISO-seq or metagenomics. So I'm very happy to introduce Gerrit guest as a guest today to talk you more about the Hi-Fi. The Hi-Fi reads provides a new lens to interrogate biogenomes and biodiversity. Where is Gerrit?
Okay, so just a short introduction to Dr. Gerrit Kohn. He's a Director of Service and Support, EMEA, representing Pacific Biosciences. He has all different roles in the same company, including Manager Field Application Specialist, Senior Field Application Scientist, Field Application Scientist, and he holds a PhD in Molecular Genetics and Mycorrhiza from the University of Lausanne. Please welcome Dr. Gerrit Kohn.
Okay. Good morning, everyone. Thank you. It's my great pleasure to be here. As I was just introduced, I'm speaking here on behalf of AGBL and Pacific Biosciences. And I realized listening to my speakers this morning that maybe many of the things that I'm going to tell you now are going to be seeming like science fiction, but I can assure you it's not science fiction. It's possible, it's real, and you can really do it. The focus and the goal of my talk is, up to the workshop that we're having here, is solutions for precision genomics and biogenome analysis. And what I'm going to do in the next 20 minutes, I'm going to take you very, very briefly to our company and our technology, but I'll not really focus on that. What I want to focus on is really the things that you can do it, the applications that have now become available thanks to our technology. And so I'll start with a very quick introduction of PacBio itself. The company has been around for about 10, a little more years, actually, the company started to bring the first commercially available sequencer to market in 2010. And the latest generation that has just been introduced by Islam is seen on the right-hand side. So you can see there is the review system. If the pointer will do it, it doesn't matter. On the right-hand side, you see the review system, you see the ONSO system in between, and you see the Sequel IIe. The Sequel IIe is our established platform and around since some time. The other two instruments have been launched last year at ASHG. And with these systems, what we're providing is a complete solution. So you see on the left-hand side, on the one hand, we have the consumables that come with the systems, and they take you from sample extraction through to library prep to actually loading the sequencer. And our instruments come with a software solution as well. And that means not only the calling of the bases, but really all these new applications that you're going to hear about now, they come as part of our software suite. I'm going to briefly focus on that as well. Let's focus quickly on these two systems. And it's truly exciting to say that we are the first company to be in a position that can offer you both long reads, native long reads, and short reads. So what we can do as a company, what AGBL can do as a company, is we can come to you and ask the question, so what is it that you want to do? We can either serve you by having a complete solution for long reads, or we can have the short reads, or we can have any kind of mixture of both. So that means you're not limited in any way by what you want to achieve scientifically. Okay? What we've done predominantly with the Revio compared to the Sequel II is we've increased our throughput and we've driven down costs. So we've heard this morning, this has been one of the major things that Illumina managed to achieve, and they're tough competitors in the field. And I have to say, we have to stand up against this, and this was one of the major obstacles we had to overcome. With Revio, we're feeling we are much, much closer to where we need to be. So with this system, you are in the position to sequence up to 1,300 genomes in a year, and you can choose at which speed you're going to do this. The system has 4 positions to load what we call smart cells and then run it. And a run of a human genome, of a complete human genome done by long reads, it's going to take you 24 hours. Okay. The ONSO system is not going to be my focus for today, but the ONSO system has been brought to the market, is currently being rolled out. And the system delivers between 400 and 500 million of reads. So that's kind of the mid throughput range for Illumina. But it does deliver something which is really outstanding. It allows you to have higher accuracy than what you're used to. And that's actually true for both systems. So let me take you through this. On the left-hand side, you see the basic foundational principle for long-read sequencing that we have. You take a piece of DNA, you ligate hairpin adapters to it, and you load it on the instrument. And on the instrument, what happens is, actually, I can use that here. On the instrument, what's going to happen is you can have your molecules diffuse in what we call these tiny nanowells, zero-mode waveguides, we call them. And they allow for a single molecule to go in. The molecule gets attached, and the sequencing reaction, as the polymerase, which is the enzyme that is bound to one of those hairpin adapters, is going to incorporate nucleotides, and they're fluorescently labeled. And each base that is incorporated is filmed. So it's not a picture, it's a movie. And the difference is, this way, we can not only see the way, actually the actual base identity, we can see the way how these bases are incorporated. And that in itself allows us at the same time, without any extra reaction, to look at the epigenetic information, because the native DNA is going to lead to a different incorporation pattern. And we can see that. So what we do at the same time is we actually have this molecule sit in a tiny ZMW, and then we're going to monitor the reaction for as long as the polymerase is active. And usually that means for fragments of a size of between 15 and 20 kb, these are the hairpin adapters, we can go around several times. And the unique feature is that you can pass on the same DNA molecule multiple times and call a consensus. And that allows you to get to an accuracy, which is the standard consensus accuracy that you would get from Illumina. But what you have here, this is a single molecule. You will go for a 30x coverage on your genome, and that means you can build from that accuracy. So we're not finishing at 30 or 40 QV, we're actually starting there. Where we're ending is rather a QV score of 60 or 70, and that's the science fiction part, is true. So let's go through that. Who is using that technology? So one of the most famous users that we have is in the UK. It's the Wellcome Trust Sanger Institute. And I'm saying congratulations here. About a month ago, they posted on their website that they completed the first 1,000 genomes of one of their hallmark and landmark initiatives, which is to sequence 70,000 endemic species of the UK. The goal is to establish high quality reference genomes. And this is exactly the spirit of this workshop, I suppose, that you want to conserve your biology, your nature in Africa. And this is one of those initiatives that proves that our technology has a place in exactly that niche. So the 1,000 genomes are publicly available. They're being done by the Wellcome Trust and made available for anyone in the scientific community. And that's a real reason to celebrate. So there's 3 parts of my talk that I'm going to cover now. Firstly, I'll briefly go into DNA extraction and library prep. I'll then focus on the sequencing, the different applications that you can run, and I will finish with some. I can't go into all detail and everything with just one example of the data analysis, but trust me, there's more. So go through the sample and library prep. So for sure, you can take a single sample, extract your DNA, put the hairpin adapters on, shear the DNA, put the hairpin adapters on, load it onto the sequencer. That's very easy. What we have to do now with the arrival of Review is we have to worry about how can you process many, many more samples. So we've been looking at a certain type of workflow to really automate. And what this workflow needs to have is it needs to be scalable. You will not have to fill the instrument all the time. So you want to run in batches at times. You want to scale it up or down. Ideally, the workflow would entail everything to be automized and automatable. You want to have the same high quality as if you would do it one by one on hand. And of course, you want to contain your cost. And the way we achieve this is by actually collaborating with an industrial partner. We've chosen Corteva Agri-Science to do this. They're based in the US, and they sequence through large accessions of plants. And so they're very familiar with automating workflows. We've partnered with them, and you see the 2 announcements here. And what has come of that collaboration, has been this workflow. So as I mentioned, we take it from DNA extraction, which is part of our workflow, and that has been automated first. And we do this on this example with a kingfisher, but we're validating more systems. And that allows you to run at a minimum a batch of 8 samples, but you can go and do more up to a number of 96 samples, always in increments of 8. And the same has been done for the library prep. So we have the Hamilton system here. And again, we're looking at more systems, but our kits have been established and adapted. And we have a protocol available with Hamilton that allows you to automate this. So you can run up to 96 library prep reactions at the same time. And finally, the system has been introduced. So we have the Revio sequencing system. So in 24 hours, you can do the equivalent for human genomes in 24 hours. Depending on the genome size, this would give you 30x coverage. You can choose another organism. You can have larger organisms or smaller organisms, and you would start to either multiplex or actually build the data for a much larger genome. Let's talk about the sequencing. Again, that part might be sounding like science fiction. The reason that it's not science fiction is you can see this has all been published. One of the things that I wanted to do first before going into details is Sequel IIe has been the first instrument to deliver what we call those Hi-Fi reads, those high quality long reads of 15 to 20 KB. And we had to prove that the Revio system does the same. And these are the statements that you see here. That is actually the case. And if at all, actually the Revio comes out a little stronger in comparison to the Sequel II. So we can say from a quality perspective, we're in a very great spot. Now, as I said, what it does is it delivers you more data and at a lower cost. And that's the particular difference with the system. And as I mentioned, it might sound like science fiction, but we can simultaneously and without any extra work record 5-methylcytosine and 6-methyladenine. 6-methyladenine is the publication here, but I'm going to show you data how that looks like. So let's go into a little more detail. What is it, if you want to build a reference genome, that you really want to do? It won't stop there. You will have this high quality of reference genome. What do you do next? You want to publish. You need to annotate your genome. That's one of the first things that you do. And for doing that, we have an application that I'll introduce in just a second. At the same time, you might look for something that is genetic, but you're not sure. So while you get the picture of the whole genome, you want to look at the other layers of information as well. And you might want to look at your epigenetic level. And that's what you can do with the system as well. And finally, you might work on an organism. You have different organs, you have different tissues. And thanks to the single cell technology, you can extract the DNA from single cells and then take a detailed look and build a cell atlas. So I'm going to go through the publications that we have, a small subset of publications that we have at this point. So iso-seq is our way to call RNA-seq. Iso-seq is called iso-seq because what you get when you do this, you take your RNA, you convert it into cDNA, then you prepare it as a library, and then you load it on the sequencer. And what it does, it simply is a catalog of all the different RNA molecules that you have extracted that you will sequence. And you don't need to break it up because the molecules are smaller than what we can take on our system. I mentioned 15 to 20 kb is easily what we can sequence. RNA molecules are typically smaller. They're 1.5, 3 kb, maybe up to 6 or 7 kb, but not much more. So iso-seq has been used extensively. And what you will have at the end of such an iso-seq run is you will have a transcriptome reference. So a complete catalog of all the isoforms that are part of your RNA. And that's why we call it iso-seq, because you get a catalog, complete collection of all the isoforms. And what you can do with it, you can annotate your genome. You can map it against the genome. You can find back the genes that have been part of your DNA sequencing. And you can take it to the extreme by employing things like the 10x instrument. And this has been one example, and I want to highlight that has just been published this year in September, that really took it to the highest level to look at what is it in a human genome that can be found on an isoform level. So the different tissues that are shown on the picture, and the different tissues here, RNA has been extracted from single cells and has been sequenced to great coverage. And there were real strong insights. You need to imagine that you've been sequencing the human genome or other species since years, and you would never expect major breakthroughs to come from that because you would think you've had a good insight into this, and that's not true. What actually has been seen here that we have had 200 full-length transcripts, and about 40% of them contain junctions that have not been observed before, novel isoforms. This is groundbreaking. And at the same time, what you will have is, we had the dogma almost to say that there's always one predominant transcription isoform that you find. And that's not true either. By actually going with little amplification, having a true representation of your transcriptome, it was found out in this study that about 73% of all the genes had several predominant isoforms. And that really has implications on how you interpret your data. So given that RNAs are small, there is something that you can use and leverage to make the sequencing that you do on our system more effective. Imagine your RNA molecules are 1.5, 2 kb in size, but you've got a length that you can sequence. Excuse me. You have got a length of sequence that you have about up to 20 kb. What you can do is you can take your library molecules and you can concatenate them. You give them a unique identifier, and then you do this reaction where you ligate them all together, and in this case up to 16 times, and that way you take advantage of the full length that can be run on a system and you increase your throughput. And that really allows you to get much more insight into your transcriptome level and go to even smaller, frequently present variants. I'm going to take you on a journey now. So this is the unit that we use for sequencing. We call it a smart cell. This is here in the center. It's like a square centimeter in size. And what you can do now with this, as I said, without kind of any extra work, is you can run on the same run, you can obtain 4 genomes. And this is quite amazing to be able to do. So let me take you through that. There is the first group that has done and employed this, this technique. And the publication, again, came out this year only. But let me take you through this. So what have they done? They've actually turned our system from a 4-base sequencer into a 5-base sequencer and eventually a 6-base sequencer. And what do I mean by this? So you can expect for any kind of DNA sequencing, A, C, G, and T as the DNA letters that we're all familiar with. As I mentioned, since we're taking a movie of the incorporation events, native DNA with a modification will lead to a change on the way the nucleotide is incorporated. And we can see that. And since every native modified base has a different pattern, we can distinguish it. And that allows us to detect 5-MC by looking at the kinetic information and 6-methyladenine by looking at it again. And that's what has been done here by this group. So what they've done is they've done ChIP-seq. So they've extracted the DNA with the nucleosomes. Knowing that the accessible part of the DNA is here, they've used the methyltransferase to transfer a methyl group on the adenine, knowing that we can detect it. And that means the parts that have been protected by a nucleosome were inaccessible, no modification occurred. The parts that have been open have been modified. And you can see that when you do the sequencing, firstly, normal sequencing strand here, but then you look at the presence of 6-methyladenine and you see in all those pink or violet color, you see those parts that have been non-protected, open, where bases could be modified. And so you can take a look on the presence of nucleosomes. And a more complete picture of this is shown here. So you can see here really the regularity when nucleosomes have been present. And you can see here as well, when transcription start factors bind to the DNA and when those regions are open as well. So you can really make an assessment of the structure of your DNA. So I've told you about RNA-seq, taking RNA, converting it into cDNA, and then putting it into a library. I've told you about DNA sequencing. By doing this methyltransferase reaction, you still have all the 4 letters available. You still have access to all the 5-methylcytosine, but you get now access to the chromatin structure as well. So you can simply take all of this on the DNA as one reaction. You can take the RNA, a small amount, as the other reaction. You pool it both together, and you can sequence it with the same system. And that means you get 4 omes with the same sequencing reaction. You get the ChIP-seq genome, you get the epigenome, the 5-methylcytosine genome, you get the RNA transcriptome reference, and you get the genomic reference. Okay, and how does that look like when you do this? This is from the publication. So firstly, the principle has been shown here. You can see the different levels here. And you can also see here on the bottom is the transcriptome reference. Here is the epigenome reference on the top. And then you can see the ChIP-seq part as well. Firstly, you can see the 2 different parts here. This is when you do it in a complete phased fashion. So you have on the human genome 2 alleles. Animals have it frequently as well, mammals, but then you have different situations. It doesn't matter for us. Given the accuracy, you can unambiguously distinguish between the 2 alleles that you have, or the several alleles that you have available. So this is one of the groundbreaking news with this technology. You get the DNA information, but you get the phasing information, and you get these other levels of layers of information. And if you take it really to the extreme, shown in the next picture, you can see here the 2 alleles, the parental and the maternal allele. You can see the differences as displayed here by the small nucleotide variants in high accuracy without errors. You can see the epigenome here. You can see the chromatin structure here. And you can see the transcriptome reference. And it is all done with a single SMRT cell. Now to complete, because what this is we've squeezed a lot into one of those single SMRT cells. You can now, you need to analyze the data. And of course, we don't want to let you alone on this. So there's a lot that comes with the system. Firstly, we have a software called SMRT-Link, which is depicted here on the left. And the software gives you access to SNPs, to small indels and deletions, and to large structure variants. And that comes with the system as it is. What you get on top is you get the tools which are shown here. You can analyze tandem repeats. You can analyze segmental duplications. You can get the full phasing information, copy number variation, and methylation. All of this is available as well. And it's actually sitting in a different public database and is curated there. We're taking bits after bits to then integrate into our software, but this has been the start, and these things are still coming. So I hope that with all of this, I've shown you that we have a lot of tools, but I want you to believe. And in order to believe, I want to show you a couple of principles or things. So firstly, the concept that we have is, look at tandem repeats. You can have different motif sizes. You can have triplets. You can have a real motif like shown here. And you can have large things being put together. And that sometimes in hotspots of evolution is coming together with an organism and drives evolution. So these are short tandem repeats. These are variable number tandem repeats. And these are macrosatellites, and these are different classes. So sometimes when you sequence and you look at those loci, it can look quite messy, like this. But it's actually not messy, because if you look in detail, you know, the deletions that you observe here are the same, they're present. The only thing you need to do is you need to sort that properly. But normal alignment tools that you would use wouldn't be able to do that. So we had to develop the tool. And what we've done, the principle is quite simple. For those regions that are highly repetitive, we're going to look for the flanking unique sequence. That's our anchor point. Then we're going with the software into this, and we're looking for the repeat motifs, and we're going to classify them. And then we're looking at the length of each of those motifs. So the principle is shown here. You might have different motifs of different length, And once you then run it through this pipeline, you can all put it together and assemble and get a clear picture. That's part of the tools that we have. Of course, there's more, and I could spend more time. You can look at pseudogenes, you could look at paralogs, you can look at so many different things in the genome, and they're really becoming clear thanks to smart sequencing. So I hope I was here in the last 20 minutes to convince you a little bit on the potential of the technology. And again, I'm saying this is not science fiction, this is true. And I would like to finish with the mission statement that we've had. So our company has decided to really do all of this to enable the promise of genomics. And as we said, you know, it was first time sequenced in 2000, around the year 2000. The true complete human genome sequence only appeared 2 years ago in 2021. And that means it had to have another technology to fill the gaps. We only had 87%, but it's predominantly the long read sequencing that drove this to become complete. So what we're doing by committing to human health is, of course, we commit to this planet. No human health without good health on this planet, on the plants and the animals and everything that surrounds us. So PacBio is committed to driving all of this. But the anchor point for us is to enable on the promise that we've always seen from genomics and to deliver a technology that allows you to have that. Voilà. With that, I will finish. Thank you for your attention. Thank you.
Thank you, Dr. Kuhn. Dr. Islam, if you don't mind, could you please join us? Okay, so thank you all for your outstanding presentation. So we would open the floor now for questions and discussion. So it's impressive to see how much progress had been made in terms of technology in this space, not only in terms of accuracy, throughput, but also in terms of reducing the cost and making this technology affordable and democratizing technology for developing countries to be able to do this kind of research and use this technology. So I would open the floor for questions. So please take advantage. We have technology leaders here in this space, so ask them all the questions you can.
Hello. So first of all, I would like to thank Professor Badawi for his invitation and his quality of organization of this event. I also want to thank our speakers for their insightful presentations. My question, or rather my curiosity, we have witnessed the huge advances in improvements in DNA sequencing. I mean, since the early 19s, we went from a pouch of single cell organisms to whole genomes of more complex organisms such as mammals and birds. I was asking myself, wondering, does the storage technology that we possess today cope with our sequencing technology? I mean, if we decide to sequence every single genome of all the living organisms on Earth, will we have a storing technology that would allow us to store all these data? Thank you.
Was this question
for all the speakers, also for Professor?
Oh, okay. All the speakers.
It's an open question.
Yeah, the question is vast, very vast. Of course, our aim is to sequence all species on this planet to know better how we can use all these diversities. That's the first point. I would say the capacity to sequencing is not a limitation today. Just we have demonstrated in the last decade that we have developed technology and instruments that are able to I would say sequence a huge number of individuals per machine per year. I would say the challenge is much more I would say to build up the project, to sequence, to annotate, and to store the data. I would say the data analysis and the data storage and the data access will be the main challenge in the future, more than the development of the capacity to sequence on a large scale. Illumina demonstrated on the microarray in the last decade. Now on the next generation sequencing with the Nova 6 series, we can, I would say, sequence thousands of individuals per year per machine.
Yeah, maybe. Thanks for the question. It's a big question. Of course, it's something that personally drives me as well. You know, you'd like, now that you have access to all of this, how can we make sure we can get all this information? One point I want to make is you see currently the review system. And if you look on our website and you look at the comments and commitments of our CEO, we've actually committed to an even higher throughput system. And the goal is always, like our competitors have done, to lower the cost, to lower the barrier to be able to sequence everything. And so I think you will see in the future that not only could we do it now, but it would cost us something, but the costs are going to come down. So that's in your favor. The second thing is, with the access now to complete genomes, what has started, people thought in the beginning for any kind of new species that we need to sequence it once. And once we have the reference, the only thing we need to do is run arrays or run next-gen sequencing to just get the information. But that's actually not true, because many of the places that are inaccessible to next-gen sequencing or to arrays, unless we know it, are missed. So what you need to do is actually, you need to build what we call a pan-genome reference. And there's a number of consortia. There's a bovine pan-genome consortium that has just formed, of which we are part as well. There is a human pan-genome reference consortium that we have. And these consortia now help us understand not just the species on the level of an individual, but really the species on the level of its ensemble of all the different individuals. And I think that's what we're going to see more, and that's going to help us to understand what really drives species. Do we need to sequence, and do we want to sequence everything? Of course, but maybe that's not needed by understanding better now the pan-genome level that we are establishing at this point. And I think that's the breakthrough that is currently ongoing. We will have a much better understanding of evolution. We will have a much better understanding of what drives evolution. And that will certainly help us to understand what we need to do on the biodiversity level.
I just want to add one point. I recognize the scientific interest of the pan-genome approach, but just, I would say two comments. The first one is 87% of the human genome has been achieved in terms of scientific discovery and knowledge without the long-read technology. So that's the first point. And the second point, and I go to the geneticist. For me, the most important thing at the area of genomics, this is how do we collect phenotypes? Because it's important in terms of evolution to know all this, I would say, pan-genome approach. However, to transform this genomic information into an agricultural tool on a daily basis in Kenya, in Morocco, in Nigeria, in South Africa, it is how do we connect this variant to a heat detection, to the heat tolerance, to the milk yield, to, I would say, udder quality for a goat, or a dairy goat, or for a dairy cow. That's something which is today, for me, in Africa, a second challenge. How do we collect in an affordable way phenotypes which are reliable and with economic, I would say, impact?
Thank you very much, Xavier. So I have two questions, one from Illumina and Megaplex, and second one from AGBL and PacBio. So just I will start with when you end. I guess for phenotyping, artificial intelligence now is a good way to do it. I mean, using sensors and all for plants and animals, and they think it's very, very promising to get phenotype, and the technology is decreasing and decreasing. So it's very, very promising for the phenotyping. So Xavier, you have and Musab, you have mentioned specificity of breeds, animal and plant breeds in Africa, and the need to sequence to discover new variants. Just I got a question. If we come back to human, because you are working in human too, the most chip of diagnosis of cancer and all are based in Caucasian, non-African countries. I mean, and they are very used here. Does it mean that we should give it a second look to all this chip of diagnosis of many, many cancers that are used in African countries? I mean, is that something that has to be done at the scale of the country to update this chip and to include the new variant of the local population? So, and second, continues with the questions. Of course, this consortium like AfricaBP, the goal is to make an excellent center of genomics, so to gather all the power so we can have machines and all we can sequence. And this is among the biggest goals of this project, AfricaBP. Waiting for that, we hope that it will come in the near future. My question is, there is any possibility that Illumina or even PacBio make an effort of service, not machine, because we have no service here in Africa to sequence, to genotype, and to all. Often we need to do it outside, and it's very, very complicated to transfer the samples, to transfer all. So it's very, very complicated. So this is my question. I have a question for AGBL and PacBio. So I was struck by the information you gave that 73%, you have seen that 70% of the genes have more than one dominant isoform, which is really striking as the information. My question is that it was done in which study? I mean, large scale study, or because it's game changing, I guess, if you advance this information. And second, I was stuck too by the Revo Smart Cell to do all the omics. I would like to have an idea about the cost of one sample today. So this is my question. Thank you.
Thanks, Wabib. So the first question is about the backbone. You're completely right. Less than 2% of the human of the data used for human research in genetics have been done with African ancestry. That's completely real, and that's why Africa, as a Africa consortium, has been built. That's the first point. So that means these chips, for this association, all these different studies, used a dedicated tool. However, there was some gap, especially North Africa, Nile Valley, the Congo region. That's why some whole genome sequencing at a high depth has been implemented, is ongoing to be done together with Illumina to fulfill the gap, and to be able in the next in the near future to develop the third generation of tool, which will be much more precise in terms of backbone covering because one thing which is very important is African population is very different for the Caucasian. But within Africa, there is a huge diversity between here and Kigali and Cape Town in terms of type of sub-ethnicities. And that's why there is a need to really fine-tune all this diversity, to capture this diversity very precisely. That's the first question. And we have a lot of national projects in South Africa to sequence at a high coverage to tackle this diversity, and then to implement, thanks to this new data set dedicated for region, some first tool for precision medicine like pharmacogenomics, precision polygenic risk scores, and so on. And that's the first question. The second one is about servicing. Do you want to answer? First point, we are all on the same line. We want to keep the samples in Africa for many reasons. Not only for Nagoya Protocol, to respect the Nagoya Protocol, not only for that, but as well for the turnaround time, the risk of loss, the risk of, I would say, and to build capacity. That's the first point. The second point is, as you mentioned, there is, I would say, some instrumentation now on the continent, thanks to different the different projects implementing the last 5 years. So there is a way to sequence or genotype on the continent African samples. Now, how do we put it in action with the servicing? I think it has to be discussed together with AfricaBP and our local, I would say, representatives, so channel partners. How do we make that feasible at a, I would say, comparable cost with, for Europe or US and so on? That should be, if there is a will, there is a way.
Okay, I'll try to remember the questions. You need to tell me if I missed one. I think the first one, I will take that as well for the service providers. This is really important. I mean, the technologies should become available to anyone here in the room and to anyone in Africa. And we have one program that I want to call out on behalf of Pacific Biosciences. So we do have a program that is called the Certified Service Providers. And that is a program where we're going to make sure that anybody who owns a machine can make his service available, and then people can go to this lab and can expect that this lab runs with the expected quality to deliver the promises that we have made. And so we have that program, and I'm fortunate to say, actually, we have applications from Africa to do this. Our technology is starting to spread in Africa, and you can always go to these labs. You would find them on our website. Now, if we can collaborate on a larger scale, that will depend on our partners here in the region, and there's actually a number that we work with, and we would, AGBL would be the first port here, and you can go to them and actually start the conversation. And I don't know to what degree those conversations would develop, but certainly that's something we could engage on. I think the other one was on ENCODE, on the fact that you were stuck by the several predominant alleles. So I'll totally switch gears. So I haven't read the paper in depth. It came out. But what I know for a fact is that we have seen multiple times, not just as part of this paper. So this has been an intensive study on a single individual. So that's not been widespread. But what has been done is there was intensive sampling on an individual to really obtain positional information by looking at single cells of tissues and within the organism to understand how is this all coming together. This would have to be done with several individuals, but it's the ENCODE consortium that has done this. So it's not nobody. And so what they have found is consistently is that certain isoforms, because of bias that are always in any technology, haven't been seen before. We complement some of those that prevented them from being seen before, and we see them now. And because you shear your RNA, what we have done as a major difference is we don't shear it. We have it as the molecules as they are. And as we take them as they are, we get to see what there is. And because of the pretty absent lack of bias, we can actually make a very good prediction. And this is not the first time we've seen this, that we need to break a dogma. But we can assume now that hopefully there's not much more coming yet with yet another technology to add to this. But certainly there has been a layer now been added. And that was based on this individual, on this first effort. And the second thing is with the many genomes that you can do on a single cell, the cost, exactly. So I can't speak for our distributors who represent us in the region, but since I'm responsible for the European region, what we have the consumables cost that we relay. The smart cell costs about $1,000, including the sequencing reagents. Okay. On top of that comes the library prep. If you were to take all of this, what has been done in this paper, and build that, you would probably end up for one individual with a cost of about $1,500 if you were to do this in US for obtaining these 4 ohms at the same time. That's about the cost, just to give you an indication.
Okay, we have a question online. So if you don't mind, please go ahead and present yourself and ask your question.
All right, thank you so much, Professor Boabed and everyone. My name is Thangot Ebenezer, and I'm calling from Cambridge. So I can't see I can't see the paper on the screen. I just wanted to expand on the point Professor Boabed raised, because that is the next direction where that is the next direction we're kind of moving into. Quite recently, we published the paper in Nature Biotech on how will all of this evolve, particularly when it comes to sequencing in terms of building capacity and the rest. And we try to think about the center of excellence in biodiversity genomics. So from next year now, we'll begin to talk more broadly and more intensively in that regard. How do we bring institutions from, for instance, in North Africa together and try to make a case for that? And of course, we'll begin to see how that would work. But I think right here, it would be nice to again see how we can potentially link up, especially with the technology partners, and that relates to the question Professor Boabid raised on how we can actually come together, the public-private partnership, which we mentioned in that paper, to kind of form this strong, formidable center of excellence, which we believe would help us to deliver on this agenda. So I think for me, it's more of about a comment and supporting Professor Boabid's talk. And from next year, we'll move to that conversation, because such centers of excellence will now be responsible for delivering this workshop on a larger scale. We plan to have 5 of those, one in North Africa, one in Southern Africa, West Africa, and also in Eastern Central Africa. So it's just more about a comment rather than a question. Thanks a lot.
Thank you. Any other questions? Can you please come forward, if you don't mind?
Thank you. Thank you. I have my name is Bulbul Ahmed. I am from African Genome Center at UM6B. So I have 2 questions. First question is for Dr. Garrett. So in your presentation, you have shown about the single cell analysis and also sequencing of the chromatin. So my straightforward question is, does PacBio has any system or the package for ATAC-seq or DAP-seq, I mean, assay for transcriptose assay, chromatin sequencing, and also DNA's, DAP's sequencing? Does any PacBio instrument compatible for that, or any package or any system to do this sequencing?
Can you repeat the first part of your question? I don't think I completely got the ATAC-share.
So in your presentation, you showed about sequencing of the chromatin part, if I remember that 5 or 6, I think. But there is another part of the sequencing, it's called the ATAC-seq, assay for transposes assay chromatin sequencing.
Yes.
And there is another one, DAP sequencing. I don't remember the full name right now.
Yes. So you'd like to have it explained again?
Yes.
Okay. So the way this occurs, you need to imagine when the DNA polymerase sits in the ZMW and it incorporates a nucleotide, while on the reading strand where it sits, there is a motif. The speed with which this nucleotide is going to get incorporated will change. The molecule, the DNA is like a tunnel, and the DNA gets pulled through. So some signatures are like it hits the tunnel of the polymerase once, and then instead of going to the next nucleotide to incorporate, it takes a break. So there is a delay between incorporation events that we see, and we can measure that. That's milliseconds. But since this is single molecule technology, it's actually very sensitive. We can measure that and then identify, okay, the DNA is pulled through the tunnel in this way, and then we know there is a 5-methyl cytosine that we see. The 6-methyl adenine is on a different base, so the context of the bases around this is different, and also the speed with the bumps that we encounter are going to look different. So if you look at the kinetic profile, it looks like different mountains that you look at, and the mountains are the moments, we call them IPD, interpulse durations, where the DNA polymerase stops for a fraction of just a few milliseconds. And we see this repeatedly and consistently. And that's how we know that this is a 5-methyl cytosine, this is a 6-methyl adenine. And actually, we could even see more modifications. We just need to train our system to understand the way, the moment the DNA polymerase pulls one of those modified DNA molecules through it, how that actually leads to a pattern. As soon as we see the pattern, we can identify it. So that's how this would work.
Okay, thank you. My second question for Musab and you, both of you. So I'm interested for like the single cell genomics. So as you have shown a little bit, And we know that mostly for single cell genomics, it's highly, it's done in the cancer genomics or the human genomics part, but very barely done in plant on microbes. So I work with the microbes. So, and there are, when we do the sequencing, doesn't matter with Illumina or PacBio, you know that it's a huge number comes as an uncultured or unclassified bacteria or fungi. So in the end, we need to get rid of a lot of data. We cannot do anything. But recently, some people started doing the single cell genomics analysis. So does Illumina or PacBio has any compatible system with flow cytometry or the microfluidics that can directly go and sequence thing can be applied for the single cells for any microbes or fungi?
Thank you, Bulbul, for the question. From our side, we really don't know if there's a solution for single cell coupled to sequencer. I don't think Xavier also, do you have an idea about that? No? No.
Not at this point. No. No, we wouldn't have a direct solution available either. Sorry.
So we have to either use the flow cytometry or microfluidics, and then we can take it for Illumina or PacBio.
Correct.
Thank you.
Thank you. Any more questions? I would actually ask a question. This is maybe for Xavier, but everyone is welcome to answer. I'm just curious, since you have experience in the breeding industry, using these genomic tools, how do you see including climate change and the fast pace that we are seeing changes happening? Because in the breeding space, you are looking for generations to see the effects in terms of growth, disease resistance, whatever performance you are looking at. But how do we predict in the breeding using these tools for climate change as well?
It's a very good question. The first point is thanks to genomic selection, we reduce generation interval, because now, let's take for livestock, for cattle, you can choose AR bulls a few weeks after the birth. So as soon as the puberty is there, you can produce semen, you can use it on a large scale, which before you had to wait 5 years, the result of the progeny to get that. That's the first point. The second point is, I would say for the climate change, it's very important to see how the production, you measure when the production is starting to decrease in different environments. That's the first point. And the second point is by indirect measures with the increase of disease, mastitis especially. It's not a direct measure, but it's phenotypes indirectly that can give some information about which line is more resistant than the other. But personally, I strongly believe that the future will be crossing. And that's why to be able to, and in a monogastric species, you use the different lines and you use the complementary by crossing and use the cross only for production. It's not the case in ruminants. It may be tomorrow an advantage by using the heterosis effect, but by keeping the different pools of genes in different nucleus that can be bred as a normal, as a conventional breeding program from now on.
Any other questions? Okay, so please give a warm applause to our guest speakers for this outstanding session. Thank you all for your time, and this is now time for a break. So thank you. Hello. For the online participant, Professor Suhail Gouar, are you there? Professor Suhail Gouar, please give us a hand if you are there. Okay. Professor Bayram Jimali, are you there? Okay.
Good morning. Sabah al-Noor. Assalamu alaikum.
Sorry. Welcome back to the next session. I will let my colleague, Professor Bouhaddou, to present the speaker.
Thank you. Thank you. Welcome back to this session on Northern Africa biodiversity landscape. Since Professor Suhail Gouar is not online, we move directly to Professor Bayram Jemali, who got his PhD from Agronomical National Institute of Tunis, University of Carthage. Professor Jemali has made considerable contribution to environmental science and biodiversity conservation. As an assistant professor at the Mater Higher School of Agriculture within the University of Carthage, he combined academic excellence with hands-on research, collaborating with the National Gene Bank of Tunisia. Professor Jemali is also a fellow of the Inter-African Bureau of Animal Genetic Resources and an active member of the Tunisian National Committee for Animal Genetic Resources. Since 2019, he has served as a lecturer in genetic and animal biotechnologies. Professor Jemali's dedication to biodiversity conservation is evident, and we are honored to have him as our speaker today. Please join me in welcoming Professor Bayram Jemali. The floor is yours.
Shukran. Merci beaucoup. Thank you very much. Assalamu alaikum. Honored to be with you for this seminar and for this scientific 5 days. I want to thank all the team of African BP and also for our colleague from American Institute. And I want to thank all the other colleagues to be present to share and for networking and to work together for our African future. So my contribution is regarding the quality of DNA or PCR and how we can improve the results of sequencing program or technique. As you know, there are many errors can affect sample collection or conservation or during the transport of these samples to our lab, and also when we do our protocol for extraction, for amplification, or for storage DNA or other biologic samples. There are many sources of inhibitor that cause low quality or that can causes the degradation of the DNA and also the quality of PCR. What is the link between my presentation and the African BP project? We speak about our natural resources and especially for my case, animal genetic resources and how we can enhance and valorize what exists like local population or local breed to save the future of our pupil. So if we want to valorize, we need to do three different actions: inventory, characterization, and also editing or analyzing the results. And the first step is critical because the output of all the experimental process need to be sure that our samples and our result as PCR is good and the accuracy is good. So we need to take into account the quality of samples when we collect them, and also the quality of PCR. To enhance the specificity of PCR, we use different technology, and this technology have as a result the identification of genetic diversity, and by this way we can valorize what exists as this genetic diversity. And when I listened the discussion after the presentation of our guest speaker, I see that heterosis effect is important for our future as a population in African country. So the identification of genetic polymorphism using PCR or sequencing method is important. So for the accuracy of sequencing result, it depends the standardization of technique and also for the quality of PCR product. This standardization of quality and control serve to establish and to maintain and guarantee a high level of quality of provided PCR product. And if we don't carry about the samples or the PCR technique, we can get false positive results. And this false positive results can affect our final results and our identification of animal with higher or with moderate or with lower value. And we cannot target or attain our objective as genetic conservation or genetic improvement program. We need to take into account the quality of material, machine, or equipment used in at a different level, started from the samples collection to sequencing results analysis. As you know, in the level of cell, DNA is fairly stable. And when we collect samples, it can affect the quality of samples and the quality of extracted DNA if there are many contaminants or there are many factors can affect this quality. So DNA is a fragile molecule and it can be degraded between or when we during the extraction process. And if we don't have a good quality of DNA, this can affect the quality of PCR product. And the quality of PCR product can be or negative, I mean false negative, or poor quality. How we can verify, how we can evaluate the quality of extracted DNA. There are many methods used to do that. For example, optical density and agarose gel electrophoresis, and also the use of fluorescent DNA binding days. And these methods are convenient but have varying requirement in terms of equipment needed, ease of use, and calculation to be considered. For example, for the calculation of the quantity of DNA, the quantity of used primer, the quantity of different reagent used for the PCR amplification reaction. So this quality of DNA, and during the process of evaluation, we need to take in account the quality. When we use the agarose gel, we need to have and to verify the absence of contamination, the absence of protein or RNA in our solution, and it can be verified directly when we use gel of agarose, or when we have a good quality of DNA, and we take on account the ratio of absorbance at 260 and 280. And the ratio, it will be between 1.7 and 2. If we have this ratio different from this interval, we can explain that by the nature of contamination. And if we have contamination with the protein, we need to use the proteinase to avoid and to degrade and this contamination. And if the DNA is contaminated with RNA, we need to use RNase to have a good quality of DNA. Also, during the experimental process, we need to control temperature, because it have a high effect, an important effect regarding the integrity and the quality of DNA. DNA purity can be severely affected by virus compound as polysaccharide, lipid, chemical reagent used in the process of DNA extraction, and also when we prepare the PCR reaction and the ingredient. The major used reagent is lithium diamine tetracetic acid, LDTA, and is known as factor enhance the integrity and the conservation of DNA. So we need to have a good concentration of for that, and the ability to conserve DNA regarding the action of RNAs. It will be as at the second level, because when we use EDTA, it inactivates the RNAs, and it can improve the quality of extracted DNA. Using EDTA, we can speak about stability of extracted DNA. Also, we need to take into account the pH of solution, and it will be ranged between 5 and 8, because when we exceed this interval, we can affect the quality and the integrity of DNA, and it at first level as contain of purine for DNA, it will be affected when we are in low pH low than 5, and the purine and the pyrimide is the essential content of DNA, and the control of pH can also improve the quality of extracted DNA. Other sources of inhibitor can be cited like the used material and the used equipment when it contaminant or it not sterilized. Also chemical reagent that can an important impact during the process of DNA purification. For example, we need to take an account and to have a high attention when we use KCl or NaCl or other salts, because it affects directly the results as PCR product. Also, ionic detergent, sodium desoxy, chlorate, sarcosyl, SDS, or ethanol, or isopropanol. It's good to extract and to separate DNA from the other content of the solution, but their effect is not good for the quality of PCR, because it can inhibit the activity of polymerase, and we can have as results lower quantity and quality of PCR product. Speaking about the quality of PCR, PCR product is used to be sequenced when target a fragment of DNA or whole genome, and this quality depends to the used material or equipment, the used primer, the fiability or accuracy of target sequence, and also it includes an internal or external factor can affecting this the results of PCR reaction, polymerase reaction. Donc we need to have specific attention for the quality of used samples, the material in the machine, the methods. It need to be standardized and optimized. For example, when we speak about the melting curve or melting temperature, we need to have a good monitoring and dispatching for material in our lab, and also we need to confirm our measurement methods to be sure that we will have a good PCR product, and it will be can use it for the other step of characterization. This quality control methods are a set of technique, and it will be can use it when at the step or level of sample collection, DNA extraction, or PCR product. To ensure this high quality of results, we need to design or to detect or to reduce this source of error. We need to take to be what we have as equipment in other lab, and it will be need to be calibrated for use and for specific use also, because we don't have the same or the one type of experimental process. We need to inventor and for our reagent and their validity to avoid the use of expired reagent. Also, we need to have manual of procedure for different used technique, and this is need to be documented and shared for all the member of the lab. In order to improve the quality of our experimental process or our lab, the calibration of all used tools it recommended. And this calibration can improve the accuracy of the results. Also, we need to have a good monitoring for our samples, starting from the first step of collection to PCR product as the last step, or sequencing the PCR product as the last step. Also, we need to have a good knowledge regarding the analysis of sequencing results. PCR product can be affected by different factors, and specifically with magnesium, and also even the primer. When we use a false or not specific primer, it affects directly the results of target sequence. And also we need to use the optimal concentration and the quantity of different ingredient for polymerization reaction. So this factor can affecting the specificity and the efficiency of PCR is generally the quality and the concentration of template DNA, the design and the concentration of primer, the concentration of magnesium and other ingredient like desoxyribonucleic, and also PCR buffer. It will be specific and don't have an effect for DNA polymerase, and we need to use adequately and optimal concentration of this polymerase. We need to take also in account the number of cycle for PCR, because regarding the quality of our DNA polymerase, we can change or recommend the number of cycle to have the needed quantity of PCR product, and it can minimize the errors when we want to sequence this, this product.
You, Professor, you still have 3 minutes.
Okay, so there are many sources of error associated with DNA samples extraction and also polymerase PCR product. So I can recapitalize that when we have an attention regarding PCR product and our DNA samples, we can have a good results as a sequencing process. Just I want to share with you some results of PCR product on a jet of agarose. For example, how we can interpret this results. For example, in this case, we can adjust the TM, the temperature of used, and also the concentration of magnesium. In this case, we have a high quantity of polymerase, because we have the good quality of PCR product, but this can affect the manner of to identify the white of our product. The same test for here. We have at the same time bad temperature and also concentration of magnesium. In this slide, just to say also the quality of used gel, it can affect our manner of our decision regarding the PCR product. regarding the profile of this electrophorese, we can decide that the gel, agarose gel need to be recontrolled. And thank you very much.
Thank you so much. I ask you to wait for the question and answer session that will take place after all presentations. So without wasting more time, we move to the next presentation, which is the presentation of Professor Suhail Gawar. Hopefully he's still here.
Yes, I'm here.
Okay, perfect. So I introduce you before giving you the floor. So although Professor Gawar will be presenting remotely, his extensive academic journey and contributions to the field of genetics make him a valuable addition to this session. His academic path began in 1997 with post-graduation studies in molecular genetics, leading to a magister degree in June 2002 and a doctorate in June 2009, with a focus on ovine genetics under the guidance of Professor Saidi Mahtar. Throughout his career, Professor Gawar has made substantial contribution to research, authored numerous papers and books, and played a pivotal role in genetics education. His leadership extends to organizing international workshops and participating in national and international expertise initiatives. We appreciate your presentation and we promise to enrich our discussion. The floor is yours.
Thank you. Est-ce que mes diapos sont... Est-ce que tout le monde voit mes diapos?
Oui.
C'est bon? Merci. بسم الله الرحمن الرحيم والصلاة والسلام على رسول الله. الشكر موصول لكل المنظمين والمنظمين في المغرب الشقيقة، وأيضا المنظمين المسؤولين في مشروع أفريكا بي بي. شكرا لكم. إذن يسرني أن أقدم لكم محاضرة معنونة التنوع البيولوجي الحيواني في الجزائر والتحديات التي تواجه هذا التنوع البيولوجي. طبعا الجزائر لها مساحة كبيرة في موقع جغرافي فريد، مما جعل من ذلك تكون أنظمة بيئية متنوعة، وهذه الأنظمة البيئية طبعا هي محل لكثير من التنوع البيولوجي النباتي والحيواني، وفي الكثير من الأحيان هو فريد بالجزائر، وإذا هناك عدة حيوانات مستوطنة بالجزائر فقط. على غرار مثلا، على غرار الفنك والفهد الصياد وغيرها. هذه الحيوانات الفريدة من نوعها تلعب دور كبير في تكوين الأنظمة البيئية، ولها دور أيضا مهم في الكثير لها كثير من الدور في الثقافة المحلية. طبعا الحفاظ على التنوع البيولوجي له أهميته في الجزائر وفي إفريقيا أيضا. التنوع البيولوجي ودراسته يمكن طبعا أن يكون الحل لمعضلة الأمن الغذائي التي تسود الكثير من البلدان حول العالم، ولهذا وجب دراستها ومعرفتها. في الجزائر نجد عدة أنظمة بيئية على غرار النظام البيئي البحري، إذ أن هناك عدة جزر وشاطئ يمتد إلى 1600 كيلومتر. هناك جبال الأطلس، هناك جبال في التل وجبال تفرق بين التل والصحراء، وهناك أيضا جبال في الأطلس الصحراوي. طبعا الجزائر تتكون أيضا من حوالي 80% من مساحتها هي صحراء. في هذه بين الصحراء والتلال هناك نظام بيئي فريد يسمى بالهضاب العليا، وطبعا نجد أيضا في وسط الصحراء وعلى أطراف الصحراء ما يسمى بالواحات، وفي هذه الواحات أيضا نجد أسماك وحيوانات فريدة تعيش إلا في هذه المناطق. كما لا ننسى أن هناك جبال معروفة ومشهورة في صحراء الجزائر على غرار التصيلي وجبال الهجار. طبعا يتخلل الصحراء نظام بيئي فريد يسمى وديان الصحراء. هذه الوديان تتسم بكون أن هناك تكون مملوءة بالمياه لمدة فترة معينة فقط، إلا أنه وجدنا في هذه الأودية أسماك، يعني أسماك بهذه الأودية، واتضح لاحقا أن هذه الأسماك تلد بيضها والبيض يبقى محافظ عليه في الرمال إلى يعني تساقط الأمطار اللاحقة، وتقدر هذه البيض سبحان الله تبقى حتى 6 سنوات بدون ماء محفوظة في الرمال. وهناك أيضا نظام بيئي جدير بالذكر هو نظام الشط. نظام الشط أين نجد ملوحة كبيرة جدا، إلا أن بهذه المناطق الجد مالحة نجد هناك أن حيوانات ونباتات تأقلمت مع هذا النظام المالح جدا، وقد تكون الحل دراسة الوراثة لهذه الحيوانات والنباتات قد تكون الحل لإيجاد مشكل تملح الأراضي الذي يصيب الكثير من البلدان. طبعا هذه النظام البيئي الشط أيضا يأتي إليه الكثير من الطيور. النظام البحري طبعا كما ذكرت سالفا فيه عدة جزر، إذ أن في الجزائر هناك عدة جزر، ومعظم هذه الجزر الآن هي محميات وأصبحت الآن لا يزورها إلا العلماء أو المنظمات الغير حكومية المهتمة بالحفاظ على البيئة. طبعا عندنا أيضا ما يسمى ب الهضاب الداخلية. أين نجد على سبيل الذكر لا على سبيل الحصر نجد الغزال، ونجد أيضا كبش الجبال. هناك أيضا أنظمة ما تسمى بالأنظمة البيئية الرطبة، وهذه نجدها خاصة في شرق الجزائر على غرار طونغا والقالة. الجزائر أيضا تحتوي على تركيبة غابية مهمة جدا. طبعا تناقصت خلال الاستثمار الفرنسي، تناقصت إلى أكثر من الثلثين، مما أدى إلى فقدان الكثير من التنوع البيولوجي. إذا أردنا أن نذكر بالأرقام ماذا يوجد كتنوع بيولوجي حيواني في الجزائر، لكن هذه الأرقام طبعا كما تعرفون هي ليست أرقام دقيقة. لماذا ليست دقيقة؟ لأن عدد الأعمال العلمية والإمكانات لتجوال في مساحة كبيرة مثل مساحة الجزائر، وأين 80% من هذه المساحة هي صحراء، ليس بالسهل. لكن عندنا أرقام تقريبية. فمثلا بالنسبة للزواحف حوالي 130 إلى 140 صنف من الزواحف، من الأفاعي والسلاحف، وربما بقي حتى التماسيح. هو آخر تمساح قتل في الجزائر قتل بمنطقة جانات في الصحراء الشرقية، في منطقة جانات. آخر تمساح قتله الفرنسيون سنة 1940، وكان يعني تمساح فريد من نوعه، إلا أنه ظن كثير من الناس أنه اختفى. لكن مناقشتنا مع رجال الطوارئ تقول بأنه ربما لا يزال هناك بعض الحيوانات من التماسيح باقية في الوديان الداخلية في الكهوف، ولعل استكشاف المؤخر في موريتانيا للتمساح عندي يرد الآمل أنه لا يزال هناك تماسيح بالجزائر، لكن ليس هناك شيء مؤكد إلى الآن. فيما يخص الطيور هناك حوالي 400 إلى 450 صنف وأغلبها مهاجرة. فيما يخص الأسماك المياه الحلوة والمياه البحر طبعا حوالي 100 صنف. وهناك في المياه الحلوة الكثير من الأصناف لهذه الأسماك التي هي مستوطنة إلا بمناطق مخصصة، خاصة في الصحراء. هناك أسماك خاصة، لكن مع التلوث الأخير وكثرة يعني الساكنة بالمناطق أدى إلى تلوث هذه البرك والقضاء على معظم هذه الأسماك. فيما يخص التديات هناك حوالي 100 صنف من التديات على غرار الخفافيش. هناك أيضا الحيوانات اللاحمة، القوارض والضفريات. إذا جئنا إلى الحشرات طبعا ليس هناك أي أرقام محددة فيما يخص الحشرات، إلا أنه كثير من العلماء استكشفوا حشرات لم يجدوها إلا بالجزائر وأعطوها أسماءهم على غرار اليعسوب الجزائري. هناك فيما يخص العناكب والعقارب أيضا هناك تنوع كبير من العناكب والعقارب، لكن ليس هناك تفصيل وأرقام محددة، إلا أنه مؤخرا نحن قمنا بدراسة لجرد العقارب الموجودة في الجزائر، ووجدنا حوالي 12 صنف من العقارب موجودة بالغرب الجزائري، لكن نعرف بأن هذا العدد ليس حقيقي لأننا طبعا لم نقم بالاستكشاف 100% من الأراضي الموجودة في الغرب الجزائري. إذا نتكلم على سبيل الذكر لا على سبيل الحصر بعض الحيوانات المهمة بالجزائر، لأن لو نذكرها كلها لا ننتهي ربما مدة شهر. عندنا مثلا الماكاك البربري، هذا النوع من القردة محبوب جدا من الساكنة ويقدم عمل مفيد للبيئة إذ أنه ينشر البذور، لكن للأسف مع الحرائق الأخيرة التي جرت في السنوات الأخيرة في الجزائر الكثير من الأماكن التي كان يسكنها هذا الماكاك طبعا اختفت بالحرائق، لكن الحمد لله هناك جهود الآن من الدولة والتنظيمات غير حكومية لإعادة تشجير وإعادة تأهيل هذه المناطق. عندنا الضبع المخطط، وهذا الحيوان يعني له قصة فريدة. لماذا؟ لأنه هذا الضبع هو محل اهتمام كبير من قبل السلطات المحلية وخاصة الدرك الوطني. لماذا؟ لأنه المهربين يستعملون جلده ودمه لتهريب الكلاب حتى لا يقدر الكلاب يشمون رائحة المهربات التي يهربونها، ولهذا كل حيوان من الضبع المؤقت هو متبوع من الدرك والعلماء. وإذا مات حيوان طبعا حتى يحضر الدرك الوطني ويقوم بحرق العينات حتى لا يستعملها المهربين. وهذا الحيوان كان قد انقرض من الجزائر تماما لمدة 40 سنة، لكن ال5 سنوات الأخيرة رأت عودة هذا الحيوان وتوالده بالمنطقة، ولقد صور مع أولاده. إذن فهو الآن موجود نوعا ما بكثرة. هناك الحيوان حيوان القطب الغرير. هذا حيوان الغرير نجده في جبال الأطلس والأعداد تاعو قليلة جدا. هناك أيضا حيوان الغزلان البور. غزلان البور هذه أيضا هي في طريق الانقراض، لكنها محل اهتمام محميات كثيرة بالشرق الجزائري، والحمد لله الآن الأعداد تاعو في تكاثر، حتى أن هناك نواة مؤخرا قاموا بنشرها في الغرب الجزائري في مركز الصيد بالغرب الجزائري بتلمسان. عندنا القنفذ. القنفذ هو حيوان مهم جدا للفلاحين، لكن العديد من الفلاحين لا يفهمون ذلك، إذ أنه حيوان يقضي على الكثير من الحشرات. عندنا حيوان القطب المخطط، هو حيوان لاحم، نجده في الصحراء الجزائرية. عندنا سمك البرب، يعني أغلب الأسماك الموجودة في الوديان الحلوة بالجزائر هي من صنف البرب. والجدير بالذكر هنا أن هناك سمك برب يوجد في منطقة أدرار، في منطقة الواحات، أين يستعمل السقي بالفكارة، بما أن الفكارات هي مناطق لا يدخلها الضوء، فنجد هذا سمك البرب ليس له أعين، يعني أعمى لا يرى، ويكون يعني جد مهم لو تمت دراسة هذا النوع من الأسماك من الناحية الوراثية. عندنا طبعا حيوانات برمائية على غرار ضفدع رنطة ساريكا، نجدها في الصحراء. فيما يخص الأفاعي، طبعا دراسة الأفاعي في الجزائر هي مهمة جدا. لماذا؟ لأن كل المضادات الحيوية التي تصلنا هي ليست مضادات حيوية خاصة بالأفاعي الموجودة في الصحراء الجزائر، ولهذا فهي ليست فعالة 100%، على غرار أفعال كوبرا. يظن الكثير من العلماء الحيوان أن أفعى الكوبرا الجزائرية هي أفعى كوبرا جزائرية لا تشبه الأفعى الموجودة في مصر مثلا، فهي فريدة من نوعها، لكن لحد الآن لا توجد دراسة وراثية على هذا النوع من الأفاعي. طبعا عندنا الحرباء. الآن إذا تكلمنا عن الحيوانات الأيقونية الموجودة بالجزائر، طبعا على رأسها أسد الأطلس. للأسف رغم أن هناك برنامج لإعادة تأهيل هذا الحيوان، لكن وجوده في البرية قد يستحيل. لماذا؟ لأنه صغر مساحة الغابات وانقراض الكثير من الحيوانات التي كانت طريدة المفضلة لهذا الحيوان. لا يوجد الآن إلا أعداد قليلة منها في حديقة الحيوان بالجزائر العاصمة. حيوان الفهد ظن العلماء أنه انقرض منذ 60 سنة وسجل الانقراض تاعو، والحمد لله السنة الفارطة سنة 2022 تمكن المصور الشهير الجزائري رضوان طاهري من تصوير هذا الحيوان. طبعا الصورة التي هي أمامكم ليست للفهد الجزائري، هي للفهد الإفريقي، لكن الفهد الجزائري هو أصغر حجما والألوان باهتة أكثر بهوتا، وطبعا تم اكتشافه في منطقة حدودية بين الجزائر والمغرب.
Professeur Gouar, je m'excuse de vous interrompre. Vous avez expiré les 15 minutes, donc je vous accorde 2 minutes pour conclure.
Donc شكرا، آسف جدا على أخذ الوقت. الفهد الصياد موجود بمنطقة تمن راست. لقد حاول الكثير من الناس دراسته جينيا، لكن إيجاده صعب، أمر صعب جدا. هناك حيوان الواشق أيضا، هو حيوان مهم ويصطاد فرائس، ونجده أيضا خاصة في الأطلس. ولعل أكثر حيوان هو في طريق الانقراض فيما يخص القطاطيات هو قط الرمال، إذ أن العلماء يرجحون أنه لم يبقى إلا حوالي 20 حيوان في البرية. طبعا الحيوان المشهور ومعروف في الجزائر هو الفنك. فيما يخص الغزال، هناك 3 أنواع من الغزال، وهناك برنامج مهم ومكثف وناجح جدا في منطقة تلمسان في مركز الصيد مهتم بالحفاظ على غزال كيوفي، وهناك أيضا برنامج آخر لغزال الله المرين في منطقة البيض. فيما يخص كبش الجبال، طبعا هناك أيضا برنامج أيضا رائع وقد لقى نجاح كبير في منطقة تلمسان. في حظيره الصيد لمنطقه تلمسان، وتم اطلاق الكثير من الحيوانات في البريه. فيما يخص النعام، لقد حاول مركز الصيد بالتعاون مع جامعه تلمسان لاعاده تاهيل هذا الحيوان، لكن للاسف استيراد البيض لهذا الحيوان لم يتم بطريقه سلسه، ويعني انشقت البيوض ولم ينجح البرنامج. هذا الحيوان اللي هو حيوان صقر يتم ايضا هناك برنامج لحمايته. طائر الحباره هو طائر مشهور جدا ومفضل لدى اخواننا في الخليج، الا ان الصيد الكثيف والمكثف على هذا الحيوان كاد ان ينقرض هذا الحيوان، الا ان مؤخرا تم انشاء مركز لتوليد والتكثير من هذا الحيوان في منطقه النعامه بالجزائر. هذا الحيوان ايضا مشهور بالجزائر، نجده خاصه في مناطق الساحل، النورس ذو الراس الاسود. هذا النوع من السلحفات ايضا نجده في صحراء الجزائر. فيما يخص الحيوانات البحريه، الثدييات البحريه، عندنا الدلفين الجزائري. هل هو جزائري ام هو دلفين اخر؟ كادت ان تتم هناك دراسه وراثيه الا انها لم نتمكن منها. لماذا؟ لاننا لم نستطيع اخذ عينات من الدلفين لانه محمي جدا، واي اقتراب من هذا الحيوان هو يعني ممنوع. لقد يوجد ايضا العلماء لاحظوا ايضا وجود سمك القرش الابيض وسمك القرش ذو راس المطرقه، لكن هل هذه الاسماك القرش هي حيوانات ضائعه من المحيط وضاعت في البحر الابيض المتوسط؟ كان كثير من العلماء يظنون ذلك، الا انه دراسه قد قام بها بعض العلماء على الحيتان في البحر الابيض المتوسط، ورأوا ان الحيتان في البحر الابيض المتوسط هي حيوانات خاصه بالبحر الابيض المتوسط وليست حيوانات ضائعه. اذا الدراسه الوراثيه قد تعطينا الكثير من الاجابات. طبعا هناك الكثير من
pour cette présentation très riche de la biodiversité en Algérie.
شكرا.
Je m'excuse de vous interrompre, mais par contre, fin de temps, on est obligé de...
الله يجازيك. شكرا، بارك الله فيك.
Merci.
اوكي، شكرا استاذ جوار على هذه المداخله القيمه التي عرفتنا بها عن التنوع البيولوجي في الجزائر الشقيقه. We move now to the next speaker. So the next presentation will be called entitled "The North African Microbial Diversity Landscape from Endemicity to Indigenousness." So Professor Hijri is a full professor renowned for his expertise in mycology and soil microbiology. He has been a professor at the University of Montreal since 2005, dedicating his research to mycorrhizal symbiosis. In 2021, he took a new role as a founder of the African Genome Center at the University Mohammed VI Polytechnic, where he has served as an affiliate professor. Dr. Hijri holds a bachelor and master's degrees and PhD in biochemistry, cell biology, and molecular genetics from the University of Burgundy in Dijon, in France. He then pursued postdoctoral fellowship in molecular genetics and genomics with renowned researchers. With over 25 years of experience in plant microbiome interaction and microbial genomics, Dr. Hijri has established his own laboratory at the Plant Biology Research Institute. Through his career, he has authored over 140 peer-reviewed articles in highly impacted journals and securing over $40 million in research grants. So please welcome Dr. Hijri.
Assalamu alaikum. Can you hear me? Okay, great. So thank you for the organizers for this opportunity. It's really a great opportunity to meet and to discuss with graduate students, with colleagues. So this is my first visit here in Rabat, and I'm really enjoying my sittings. So I will be asking questions for students, and I'm expecting answers from students. And I would like really the audience to participate to this presentation. Although the time is limited, I have 15 minutes. And I will take you into a tour to a hidden diversity. So just the presentation before, Very nice animals, very nice images. And now for going really deep into the hidden diversity of microorganisms. So my presentation will be organized as follows. So a brief introductions, why microbes are important. And also I will pick 2 examples of projects that are ongoing. I will not present them all. I will just really highlight some examples of the data. And all these data are not yet published. So one paper is already ready for submission, but are new data that will be presented. And I will end by some perspectives. So let's go to our tours into, see, screen, no. Okay, here we go. So if you take 1 gram of soil, it's not that much, 1 gram of soil, you take a teaspoon and then you dig in soil. You can find billions of cells. Not I'm talking really with millions, but billions of cells. At the scale of a bacterium, if you are really taking 1 gram, it's a whole biome for microbes. And this is really just to show you this hidden biodiversity that is in soil, and soil is alive. It's not just something dirt, it's not something useless, it's very important, and soil health is extremely important if we want to really to feed the planet. So keep this in mind, how important is to talk about biodiversity of soil. Soil biota determine ecosystem multifunctionalities. This is really complex. This is really an example where you can see soil biology, how this all the individuals with microbes, mesofauna, and animals that are in soil can really influence soil processes, and all those cell processes then influence their functions. As important functions, phosphate solubilization is one example. Nitrogen fixations and all this nutrient dynamics and mineralization are important soil functions, and they are really governed by microbes. Now here, we can see all this network, very complex network that are really interacting to each other. And what I want to show with this slide, so we can manipulate some actors to improve functions. And this is something that we can well do during the day, really manipulating some important microbes to increase the system of functionality. Now, on the topic of North Africa and microbiota. So this is a map that has been published 2 months ago with some colleagues following ISMI Africa with 3rd edition that was in Côte d'Ivoire. And I mean, this is the reality. You see the data that are available on microbes throughout the planet. You see that Africa, almost nothing. You will see some yellow dots here, but most of them are really interacting, are very specific projects that they are studying microbes that they are interact with plants or with animals. But hardly you have students, I'm speaking for students, you have a lot of work to do to reach at least level for developed country. So I want not to be pessimist, but we're hardly nothing. We don't know nothing about microbiota of North Africa. And you see, South Africa, they are doing better. So there is a lot of work to do, microbiology and using tools, as I said, it's hidden diversity. So next generation sequencing, it's a must. It's a tool. You can't study this differently. So microbiologists, they are using this since decades, and this is really our primary tools if we want to really to gain our understanding and to improve our understanding of soil microbiology. Let's start with the first example, khubiza. Who likes to eat khubiza? Yeah, khubiza wala baqula and some. Only a few. How many? Yes, much better. So this is already something I just love it. So I grew up in a farm in and khubiza, it's for spring. So this is, you can't really go. So this is a dish of spring things. So the question is, why we are interested in this malva or Malva sylvestris? Was the question, our primary question was really to study Moroccan microbiome, soil microbiome. So plant is an important driver for microbial diversity and community. So in another term, if you want to do comparison, you can't compare orange with apple. So you have really to remove at least the planted driver because it's a very important factor. So let's say a postdoc started in my lab and said, look, let's jump into the car and let's go throughout Morocco for sampling a plant that we can find almost everywhere. So malva, if you see the distribution of malva, you can find it in Europe, you can find it in Western, you can find it even in China, you can find it in desert places. So it's really an example of plant in which you can see how these plants will interact with indigenous microbiota for at least to remove the plant effect. So then we decided last year, that was in end of February. So we went to tour. We didn't have really a plan. So we just jumped. We started from Ben Guerir here, and then we took road to Qalat Sragna, Bni Mellal, from Bni Mellal to going for north. And of course, our aim is really to find the population of Malva and to stop there. And local people, they were laughing for us. Oh, the scientists from UM6P, they are coming, they are crazy, they are looking for Khubiza. Whatever we can. So we have to explain them why we are interested in, and then they said, oh, wow, this is interesting. Yeah, Khubiza, I know it. And yes, they're everywhere. Please go, take it, collect it. So that started like, so 13 different sites, and each site, then we collected 5 to 6 individuals. And my message for graduate students, experimental design, it's something really crucial. Ask good questions, have clear hypotheses, and testable hypotheses. Hypothesis cannot be conditional. It must be affirmation. Then go really for, and the replications, of course, different sites, different replicas. Don't stick at 3 replicas. 3 replicas, it's the strict minimum. If a sample fail, that can happen, then you are done. Your site is not, you cannot use it for statistics. So you have to find the balance between number of replicates, but also the different sites. So these are some examples. So Jean Luget, who a postdoc at AGC, and we have here, we collected a bunch of different, different sites, cultivated sites, non-cultivated sites, a gradient of precipitation, but also different soils. So if you have different soils, then of course we're expecting that having different microbiota. You have to collect all these variables that you can use for statistical analysis as multivariate analysis. Then what we were, I know Khubiza very well, but I grow in a farm. And people on farmers, they know that there is 2 phenotypes. You have a spread phenotype. So this one, it's really spread. It has no competition around. And then you have this erected phenotype. My question is, which one you will eat? If you want to collect Khubiza for preparing a dish, which one? That one over there. On the right, erected or rampant? Yes, yes. Farmers, they know that. And my uncle was asking, farm to he said, don't touch that one. It's poisonous. Of course, if you see here, the color, reddish to pink color, don't eat that one. So it contains some toxic compounds. And people, they know that very, 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 very much. No, don't touch it. Go in the field and look for erected tank, and don't go and take that one in the edges. Go in field, and field that is not treated with pesticides, very important. Okay. So I will not really go into details of technical things, but this is something routinely done in IGC. I welcome anyone for collaboration to come to do that. We have MySeq, this is really fully running. So you extract your DNA from a sample, from soil, and then from leaves or from root, from materials. You do a PCR, this is a metabarcoding, very conventional. If you don't want to do metabarcoding, you can share your DNA and then you go for metagenome. So, and for metabarcoding, it's 16S for bacteria or archaea, and ITS for fungi. This is something really the routine that we use. And now we will be lucky to have PacBio, because we can go for the full land 16S. We have already primers Garrett, so we are waiting to install the instrument. So we can sequence at full length the 16S, which will give us the power of taxa identification. So this is something important. Then you go for sequencing here. It's really our preferred instrument, MiSeq, and then it gives our really short reads. And we are using typically V600, meaning it's 600 base pairs in two directions, ending by having approximately 40 to 400 to 400 base pairs, which is okay. Two variable domains between and before well known in bacteria. So the examples I'm showing here is just for bacteria, but we have also for fungi. So this is an overview of all the parameters that were used. I will not go into detail, but you see the different sites. You see the sites, are they cultivated, if cultivated, which crop, and so on, and so forth. So this is really important, and we will use this matrix for multivariate analysis. Before going into detail, very important, sample size. So you must do a rarefication analysis. That is something routinely done. And most of the time now, a couple of years, we put it as a figure, nice figure, but now it goes to supplementary information. But if you are using Illumina MiSeq, you will be ending by having thousands or tens of thousands of different reads or sequences within a sample, which is really not a problem. You see all the curve has reached their plateau, meaning the sample size for each, well, number of reads for each site is appropriate. Then you move forward for studying alpha diversity. What that means for those that are not familiar, alpha diversity, it means biodiversity, the occurrence of species. Who is there? That's it. What is their name? So you see here, this is two graphs, really very, very conventional, very typical at the phylum level, and then you go at the genus level. And if you look the data, that was for us a big surprise. I can tell you, I will be working with this topic since almost a decade. That was something that really popped up. Rhizobium dominated largely the roots of malva. What is the rule? What is the functions of rhizobium? PhD students or master students? I need an answer. Nitrogen fixation, nodules? Yeah. So if I'm working or if I'm studying a legume plant, I won't be surprised. But this is malva. Yes. But this is malva. What rhizobium is doing there? What is its function? I have no idea, because it's not the appropriate tool to ask here for function. So we found it there. It's there. It's dominated largely the community. And I'm putting a question. That is something that we must dig into details to understand what is its functions. It is related to nitrogen fixation. If you see Khubiza, it's very green all the time. Whether it's rampant or in the edges, it's most of the time it's green. So this is really a kind of how we create a problem, basically. Well, that problem, we have no idea what the rhizobium is doing in the roots of malva. So then we have to design an experimental an experiment to ask that question. Either you go with RNA for functions or to do some experiment of nitrogen fixation experiment. So, and also because we are really interested in understanding biodiversity, it is something that we are all the time and in having unknown taxa. It's a question, what is unknown taxa? We have no idea. Either those microbes have never been isolated and characterized, or have no idea. And you see, if you compare the biodiversity, it's after a rhizobium, basically. Yes, we still have a lot of taxons that they are all known. And that's the project, second project I will be talking about, using different approach, different methods to address that question. Here, there's some basic questions. Of course, we compare different sites. One of the questions, what is the core microbiome of malva? If you compare it here, you do some comparisons between different different biotopes, roots, rhizosphere, and bulk soil at different level. Another surprise, zero, no core microbiome, absolutely nothing. Then if you compare, another surprise, we have really just first, we have so many surprises. If you compare rhizosphere and soil, most of the time, if you look at the literature, the beta diversity meaning you are taking alpha diversity, what's the taxa they are present, but you are combining their frequency. What's their frequency? Of course, it's not a quantitative method, but it's a semi-quantitative method. So it can give us really a very good clue of their abundance. Then you add the abundance to alpha diversity. So you compare them, look at this graph, which is perfectly really grouped, bulk soil and the rhizosphere soil. Has anyone in the audience have any interpretation for that? If you compare the roots, completely different ones, which is not surprising. This is something we know very well. So the plant or the roots is a barrier that's selecting the endophytic communities or pathogens. It might be pathogens. But the difference between bulk soil and the rhizosphere soil is, anyone has any clues? No one? So that will be my conclusion. So, of course, the rhizosphere of Malva sylvestris have really a very exciting diversity that we have really documented now for ITS. The endosphere was largely dominated by the genus Rhizobium. So this is really a big surprise for us. And we have to dig what is its functions. The rhizosphere and the bulk soil here, slow, not any significant differences. And the other thing is because February last year, if you remember, that was very dry weather. During the sampling, the temperature was almost 30 to 35 degrees. And our interpretation, it's the fact that the environment at that time was very dry. So just the plant didn't really influence the community surrounding this, because probably it reduced the exudate. And therefore, the influence of the plant was not as that strong during that period. This is really the, I will say, the explanation that that we can provide. So the rhizosphere, yes, showed increased alpha diversity, which is something that is new, but much less than the other. How much time? Almost. So then I will have really to speed up for this this one, 2 or 3 minutes. Yeah. So Bobby, you will not have. So Bobby will give me some of his time. No, I'm joking. So I will speed up. What I want to show with here in this project, it's using culture-dependent method, meaning we want to go into isolation characterization for a plant which most of you guys will know is harmal. So it's a plant that you can find in arid environments. And here, 2 PhD students that are already very happy because each one working in a different plants, and their plants, they are really living each close to each other. So they took a picture this year with one Salma working with for Harmal and Khadija for Colosantus, Al-Hajja, that they are really close to each other. So they went really through the, I mean, that's Harmal. شكون اللي عمره تبخر بالحرمله؟ أو نزيدوا عليه الشبه. No? I remember when I was a kid, when I smell ريحة الحرمل, I just run away because it's so bad and it's so disaster. And if you have any يقولوا بالجنون ولا الدار فيها الجنون ولا شي حاجة عطاهم الحرمل, even الجنون cannot stand. I mean, smell of الحرمل. They run away. Why? It's because it contains a cocktail of compounds of sesquiterpene, and you are putting harmal and you're adding shibba, it contains sulfur. So then, I mean, even though they will not stay at home. So Salma really went into isolation and characterization of this microbiota. So I would just go offside. You have here, it's really a very ambitious thing. She has done a 1-year effort, and she ended by having really 207 isolates. This is really a big work, and this is really our bacteria. She also isolated fungi. And we are here again, a big surprise. We expect having more biodiversity in roots than in leaves. It's completely the opposite. And in leaves, they contain a lot of compounds. Here the data shows, I mean, Selma found more isolates in leaves than in the roots. And then what are those that are common in this microbiome? This is also really, I will say, the core microbiome that is really in roots and in leaves. So the investigation, these are preliminary results. So to conclude for this part, so identification of potential bacteria species, we have already their identity. We sequenced their 16S. Many of them seems to be a new species. So we have to dig into their genome sequencing. And by the way, the Harmal, also we are interested in its genome sequencing. It's actually running at AGC. I want just to thank all the team that was involved in this study, and also thanking your for your attention. I'm sorry for time.
Thank you, Professor Hijri. It's an inspiring presentation. Next time, I will have the opportunity to have a dish of khabiza. I will, of course, remember this presentation. And that's what's nice about science. We're applying these advanced tools to something that we see and we eat and we go around every day. So thank you. So we're going to move to our next speaker. I'm sure you all won't know him. His name is Professor Badawi Bouabid. So he's the host of this conference. He's going to be talking about landscape of genomics in Africa. So I'm honored to give a short presentation for Dr. Badawi. So he's a specialist in animal genetics and genomics with an academic career rooted in engineering, animal genetics and functional genetics obtained from Iavi Hassan II, CIAM in Spain, and joint program between University Autónoma de Barcelona in Spain and Agro Paris Tech, France, where he had his PhD. So Professor Badawi has specific expertise through his postdoctoral career at University Autónoma de Barcelona, and also as a senior researcher in bioinformatics and biostatistics at Parco Technology Badano in Italy. And since 2014, he joined University Mohammed V in Rabat. His research interests lie in the intersection of genetic diversity, phenotype-genotype interaction, and their implication for various facets of livestock production. Importantly, his research is underpinned by a strong commitment to environmental sustainability, focusing on how genomic science can contribute to more sustainable practices in livestock management. So Dr. Badawi is actually the North Africa coordinator for the African Genome Project. And currently he's conducting a project that marries genomics and artificial intelligence to develop a genomic selection scheme for dairy cattle, horses, camel, aiding to development of more resilient, productive, and environmentally friendly livestock breeds in Morocco specifically, but in Africa at large. So please welcome Dr. Badawi.
Thank you very much, Professor Denga. So, hello, everyone. So as stated in my presentation, I will be speaking about landscape genomics in Africa. I will put focus on Morocco and North Africa, and particularly on two species, goats and sheep. So we are in this period, people are calling Anthropocene, where humans are having big impact on the entire planet. And there are three big aspects on that. The first one is climate change. Second one is biodiversity loss. And the third one is food insecurity. So if we start speaking about climate change, you are all aware that the temperature is increasing, as we can see in this figure. And we are the big reason of that. I can think directly about anthropogenic greenhouse gas emission and deforestation and land usage, because we need more food, so we need to put more pressure on biodiversity. Sorry. So if we speak about food security, in this figure that you are seeing, here, there are some projections that have been done. And you can see in this figure on how much food we will need moving forward in green line till 2050, and how much food we are producing in orange line at the global scale, and in red line in what concerns the low-income countries, the most African countries. So there is a big difference between what we are producing and what we need. And this will put much pressure on the biodiversity because we need to have more food. So biodiversity will be worse more and more in the future. So biodiversity itself is being challenged. This data that you are seeing here in this figure is from IUCN Red List from 2007 to 2020, showing that the number of species that are in danger is steadily increasing. If you look at the bottom here, as the bottom, the blue one at the bottom, this is our mammals. So because the mammals is a species in which we put the most of focus. But as you can see here, they represent small percentage of all species in Earth, and all those species are under the danger of extinction. So if we travel back a little in time, exactly from 1500, we can see that the rate of extinction is steadily increasing. Above all, overlapping with the 1900, which correspond to the industrial evolution. So biodiversity in Earth is definitely worsening. We are going toward a catastrophe in what concerns biodiversity. So one way to look to all those challenges done or caused by biodiversity loss, we can do it through what we name the SDGs, the United Nations Sustainability Goals. There are 17 of them, and life on land and life on water are ones of them, of those SDGs. And of course, assessing and conserving biodiversity in land and water is a pillar for development, for sustainable development. And the next, I will take some examples of the works that have been done at a big scale in Africa, particularly in North Africa, to quantify and assess biodiversity, particularly in two species, goats and sheep. So here I will present the goat and sheep species biodiversity using a landscape approach. In this work, the work was done by Benjamin in 2021. The goal was to understand the biodiversity component in species habitating contrasted environment. So here, when some environmental changes happens, organisms respond via local adaptation, which consists of permanent change to external pressure, or they can respond by acclimatization, which corresponds to a temporary adaptation. Species, they can migrate to, or eventually they can go extinct. What concerns geneticists is the local adaptation, because this is that take marks on the genome, and we are looking to the genome of the species to detect this mark that was done by this adaptation. So the goat and sheep species in Morocco constitute an important case to study, as those species are well adapted to contrasted environment. We find sheep and goats in the mountains, plain, Sahara, plateau, and all. They have this ability to deal with different and constructed environments. So in this work, the landscape genomics was used to figure out if there is selection signature linked to the environment variation in sheep and goats. It was used too to understand if there is a congruence of selection signature in both species. And a third objective of another work was to understand if there are any geographical barriers that contribute to the differentiation among each species population. So in this big scale study, 160 sheep and 160 goat were selected in a surface of 450 kilometers square. And this big surface was chosen to cover different environmental regions with different environmental variables. So the whole genome sequencing and variant calling of DNA coming from all this animal showed a lot of genetic variability. We have 38.6 million variant for sheep and 31.7 million variant for goats. And this is a huge amount of variability. And this means that there has been no bottleneck for this species and that the flow has been always maintained between the population of those species. And this weak population structure even is sometimes not good, but in this context of landscape genomic is very good because population structure could be a confounding variable that could bias the understanding via the landscape genomics. And in this case, it's a good thing for this finding. So the whole genome sequences were correlated to 10 environmental variables, including temperature, rainfall, and solar radiation to detect the selection signature and the biological process involved in this. So here is an example of the result that we can expect to find. We have here in the left a Manhattan plot of XER CLR scores across autosomes in relation to the mean temperature in July. If we concentrate on the blue chromosome here, we can find there a missense SNP that has different distribution on different regions. If we see in this figure here, we can see that the C allele is present in the north of Morocco and the T allele is quite in the south of Morocco. And if we see in this figure, we can see that the C allele, the frequency of C allele is decreasing, clearly decreasing with the temperature. So this allele is very correlated to the temperature. So taking this variant and genes, more than 12,000 and 8,000 variants linked to local adaptation have been highlighted in sheep and goats. This corresponds to 276 genes and 294 genes in sheep and goat respectively. But what is striking in all this is that we have no common variant between sheep and goats in terms of variants, and one gene in common between sheep and goats. And most association here between the variants and the environmental variables were with rainfall and with the sun. So to answer the first finding of this study is that there is a distinguished adaptive mechanism to local environments between sheep and goats, which is striking because we expect that many adaptive behavior will be repeatable. But it's not the case because perhaps that such diversity might result for a high number of biological function that could deal with adaptation to a particular climatic situation, which is named the biological redundancy. This provides more arguments for the role of contingency and stochasticity rather than repeatability. I mean that when different animals are faced with certain pressure, environmental pressure, they can behave differently. I mean, it's not obligatory that we will behave in the same way. They will develop the same manner to deal with this situation. And here we have an extreme case because we have no common genetic genomic signature between the goat and sheep species, although these two species are very near one to the other because they live practically in the same areas and they were domesticated around the same dates. So generally, how the animals get adapted to an environmental pressure, we have three scenarios. The first scenario in the left is that animals could use different genes to produce different adaptive traits. And this is the first scenario. Second scenario is that animals could use different genes to produce the same adaptive traits, because this question of biological redundancy, because many genes, they are not specialized in one function, or they do many other functions. And this is a way of dealing and giving more elasticity to living species to deal with external pressure. And the third scenario is to use the same gene for the same adaptive traits. And if we see what happened in this study, we can see that the first and second scenario were very present, 99%. But for the third scenario, it concerns only one gene that I mentioned before, because there is only one common gene between goat and sheep species to deal with environmental pressure. So this is a very interesting study that shows, because always there is this debate between geneticists between stochasticity and repeatability, and there is either two schools that argue for repeatability, that all the living species, they have some tendency to produce the same mechanism to deal with pressure, environmental pressures. And there's another school, no, they argue that the traits or the mechanism developed depends on the context in which the animal, nature selection, and on the stochasticity, on one dominance. And in this case, clearly, we are giving more rights and more credit to the second school. To not have the same point of view, or to give another point of view, there's another study that we are performing, and practically with the same objective. And this is to detect signature selections in sheep across all the world. We divided this study into 4 regions on the world: tropical, including East Africa, mainly Ethiopia, and Mediterranean, including Morocco and Tunisia, continental, including Europe, and northern, including Sweden and other northern countries. So to detect the signature selection, we used FST and extended haplotype homozygosy method. So what we found, we identified many genes related to climate adaptation, mostly unique specific to population. So in this case, we find some specific traits depending on the climatic condition of where the animals are found. And many of those genes are linked to immune responses, which is an argument to deal with or to plan breeding program for disease resistance. The study shows that indigenous breeds have a rich genetic diversity, but it is declining, possibly from poor management. So this is a point to take in consideration when improving genetic diversity of the local breeds. The native breeds are more likely, the native breeds are very climate resistant breedings than the non-native breeds, which is genetically understandable because the native breeds, they got all the time to get all the favorable mutation and genetic background to deal with the environment that is there. So to conclude, landscape genomics apply to the whole genome sequencing. It allows identifying genetic or genomic regions linked to environmental variables. A whole landscape genomics, it allows the detection of genetic variation that are specific to each environmental gradient and including several patterns. In the first study, I showed that different adaptive mechanisms have been found for sheep and goats. But in the second study I have shown, it's not the case. Between different populations of sheep, we found different mechanisms according to the environmental context in which those animals are found. To give an explanation between the two and second study, one explanation could be advanced, is that in the first study, we take the breeds in Morocco according to the geographic localization, but we know that there is gene flow between all the species. So we cannot say that they were completely separated. And furthermore, we have many studies that show many crossing between those species. But in the second study, as we took animals, tropical animals, Mediterranean, Nordic animals, they are completely isolated, at least for a huge amount of time. So we were able to find specific genomic signature to each landscape. Thank you for your attention.
Thanks, Dr. Badawi. So we're going to open the floor for questions. So please, if you have any questions, raise your hands, come forward. That would be helpful. Okay.
Hello, everyone. I hope you're all doing well. First, I would like to thank all the speakers for these delighted and diverse presentations. My question actually is addressed to Professor Jimali, who was talking about the control quality and the sample, if I remember the title well, sample and product PCR control quality. So my question is, you know that the purity is one of the most important parameters to control the quality of a product, and more especially the DNA. So I would like to know, admitting that we get a rate or a percentage of an impurities that is unacceptable, how we can deal with this situation? And thank you.
Oh, okay. Who?
Many thanks. Masa al-Khair. Many thanks for the question. I think when we speak about the rate of purity for DNA, we need to speak about the type of contaminant. If we have contamination with the protein, we need to use enzyme like proteinase K. And if we speak about contaminant as RNA, we need to use also RNAs as the enzyme to have the to correct what we have as samples of the DNA.
Thank you.
You're welcome.
More questions? Any questions online, please? There is one, so please go ahead. Abdul Qasim, turn on your mic and introduce yourself and ask your question, please. Abdul Qasim, sorry. Hamasise, can you please introduce yourself and ask your question?
You are muted, Hamasise. Please unmute yourself.
What is the concentration of DNA necessary to realize a PCR?
BioM, you are muted, please unmute yourself. BioM?
Yeah, I understand, yes. For the concentration of the rate of DNA when we need to success our PCR reaction, it depends the type of the DNA polymerase used and also the total quantity of ingredient. But it recommended after the extraction of DNA to have some aliquot of DNA with 25 to 50 microgram per microliter, and we will be sure that we have the needed DNA quantity in our PCR as concentration, adequate concentration. So it depends on the length of our target sequence. It depends with the to the used DNA polymerase and the total volume of our reaction. And it recommended that the quantity is varied between 100 to 200 nanogram per microliter. Is we want to success our PCR.
May I add a compliment?
Yes, sure.
So, I mean, the question is straightforward. It's 1 to 14 nanograms per PCR reaction. Less DNA is better. Don't put too much DNA. And if you are working for soil DNA, this is a real nightmare for the quality because you have a lot of inhibitors, and we recommend to go through the dilutions to reduce the effect of inhibitors. So the quality and all this, the amount, it's really depend on the question what you want to do. If bacteria you want to do 16S, you don't even need to extract DNA. You go directly from bacteria and you amplify your 16S. It depends on, of course, if you want to do PacBio long reads, of course, you have to extract high weight molecular DNA with as clean as possible. Even worse, if you go for nanopore technologies, you have to have the purest DNA ever to avoid troubles in sequencing. It really depends what's the purpose of the study.
Thank you, Dr. Rijri.
I agree.
May I ask? Okay, go ahead, please.
Bonjour, je vous remercie pour la qualité de votre exposé. Question pour les 160 moutons et 161 chèvres que vous avez utilisés pour l'étude. Il y a eu différentes races, c'est interracial ou a été global. Par exemple, les moutons sardis, chamhdet, et aussi pour les chèvres, les races locales, les Ros ou les autres races. Merci.
Merci. I answer in English. So thank you for the question. So Benjaluni published already the work, so you can have it look. So we have practically many breeds for sheep. We have Saudi, we have Nigil, we have all. The same thing for the goats, because the work was interested in collecting species in different geographical positions. So for the plateau, they took Benigil. For the desert, they took the man. So they took all the breeds. Yeah, all the necessary breeds. Thank you. Yeah.
Another one for Professor Badawi. I was wondering if there is any correlation between this, the adaptative of livestock to environmental stress, for instance, heat, and then is there any correlation or negative effect on human immune response, and then the health of animals?
So the finding is that the resistance of the indigenous breed, the most genes that were responsible for this resistance are immune genes. So there is clearly a significant correlation between the resistance to the environment and the resistance to disease. So one conclusion of this work is that by selecting, I put it at a recommendation, by selecting for disease resistance, we might be selecting two for climate change. There are two correlated traits. Yeah. Correlated, it doesn't mean causality. There's an association. I mean, it could be another factor, confounding factor guiding the two.
Dr. Hijri, may I ask a question? Okay, so I find this soil microbiome very interesting from an agriculture standpoint. So one of the challenges that the world is facing is to produce more food, but we have destroyed a lot of rainforests, key biodiversity areas that we don't know what's in there. But in order to produce more soy, whatever, we had to cut the forest and produce monocultures, but that led to a lot of loss in terms of biodiversity. So I think the same is happening in Morocco as well, since we are going in intensive agriculture. So we have lost some biodiversity in terms of microbiome in the soil. So how do you think Morocco should address this regenerative agriculture, goal that we are all aiming to, using the microbiome and soil, expertise?
Yes, very important question. Actually, and all these tools that we are using, it's a snapshot. We are just taking a picture at that moment for the site. So this biodiversity is dynamic. It's really dynamic. However, we know that there's a correlation between biodiversity and degradation and soil fertility. So to avoid this, we have to make amendment organic matters. So this is the carbon source of the microbes. So enrichment of organic matter can definitively help. And also now we are speaking about carbon sequestration. It's one of the best way to do it, because when you harvest your field, you have to leave some organic matter there to feed the microbe. Don't go with chips and everything when you have a soil which is really towards the net. This is subject to erosion.
Yeah.
And this is really a very a problem in arid and semi-arid environment. So having organic amendments and also the diversification, it's a really important cropping system. Avoid having monocultures. Use the diverse system with legumes, cereals, and so on and so forth. And you have to take care of soil if you want to soil to be healthy and sustainable.
Thank you so much. One last question, maybe? Which one? Oh, you already have the mic. Go ahead. Sorry.
Firstly, thank you for your presentations. My first question for Professor Hijri. why you choose Marva for your study. And my second question is for Professor Bouabid. Do you have an idea of the percentage of or how much the diversity is influenced by climate change?
Yeah, the answer of the first question is straightforward. It's because Malva, it's
Yes.
cosmopolitan. We can find it almost everywhere during spring.
It's the
That's
only
it.
reason.
So we thought of jujube shrubs, but jujube shrubs is very difficult. I mean, to sample soil. We want really to compare different areas, but we want to keep the same plant. If you want to keep the same plant in Marrakesh and go in the north, I mean, you have to find a plant whose distribution is cosmopolitan. You can find it everywhere. So that's the only rationale behind choosing Malva.
Yeah. And for the question, how much genetic diversity influenced by climate change, maybe the question should be how much genetic diversity we need to face climate change. Because we are looking to keep or to improve genetic diversity to deal with eventual climate change and environmental pressures that could come. And when it's come, we hope that we find enough genetic viability and diversity so we can deal with this problem. So this is the relationship between the two.
Okay, thank you.
Welcome.
Okay, last question.
Thank you. I also thanks all speakers for their presentation. My question addressed for Professor Hijri. Why you choose collect the sample separately in 2 years, and we know that the climate change play a critical roles in the diversity of the microbiota of the plants.
Yeah, good question. Of course, yeah. If you want to study microbiome, you have either spatial distribution or temporal distribution. Either we can sample in the same season, so different in different seasons. Of course, as I said, microbes are dynamic, or at least immunity structure and the biodiversity are dynamics. And we want really to have to generate and to document changes, yearly changes. So that's the reason why we sampled 2 years. And also the purpose, because culture dependent techniques, but also we are using culture independent techniques. We are using metabarcoding and we are using metagenomes to study the microbiomes of Harman. So that's why if you are really sampling the plant in the temporal variation, you can have ideas about temporal changes.
Thank you.
Still want to have a question? Go ahead, please. Last one. Mike.
Let's speak up.
Thanks. Thank you for the nice presentation. My question is for Professor Badawi. When you were talking about the one common gene between sheep and goats, which scenario or which justification you are opting for to explain the one common gene?
So for the common gene CA2, between the goat and sheep that are living in the same geographical position, facing same environmental variables, is expected to be more than one gene, expected to be many, many, many genes. If we believe that development a mechanism to deal with the environmental variables is repeatable, okay, between the two species or between living organisms. But the fact that we find only one, it's strange. and the explanation we gave to that is that more likely, or you can find that in the article of the author published by Benjelloun, more likely stochasticity or randomness governs more the development of mechanism to deal with the environment variable than repeatability. I mean, that means that each time animals are faced or living organisms by environmental pressure, it's not necessary that they will develop the same mechanism, even they live in the same environmental conditions, because there is biological background, there are genes that do many things, so there are many mechanisms, but at the end they do the same thing, so there is many redundancy. So that's the explanation.
Okay, thank you. Okay, thank you so much. Please join me to thank all the speakers of this session. So as we transit to our next session titled Regional Biodiversity Infrastructure Capabilities, we are honored to introduce our first speaker, Professor Mustafa Al-Fahim, holding a PhD in molecular and cellular biology. لا لا في البروجرام لا ما زال. Okay. So as I told, holding a PhD in molecular and cellular biology from Louis Pasteur University, Strasbourg, France, Professor Al-Fahim brings valuable expertise to our discussion. His career includes role as an associate researcher at Laval University Research Center in Canada, and later as an assistant professor. Professor Fahim has authored over 90 research publications spanning various scientific topics. In 2006, he joined the CNRST, where he played a role in establishing Morocco's first national functional genomics platform. In 2016, he inaugurated a sequencing facility, facilitating research in biodiversity, genetic disorders, and oncology. Professor Fahim also serves as the country ambassador for the American Society of Microbiology in Morocco and actively participates in national and international committees addressing scientific and public health matters. Please join me in welcoming Professor Mustafa Al-Fahim as he leads our discussion on regional biodiversity infrastructure capability. Thank you.
Assalamu alaikum wa rahmatullah. Thank you very much for this warm introduction on my behalf. First of all, I would like to thank the organizer for giving me this great opportunity to take part of this outstanding event. Special thanks to my colleague, Professor Bouabid, who paved the way in front of my institution to take part and to sign this convention with the Africa BioGenome Project. So I really express my great gratitude for this facilitating. So I really and I sincerely want that our institution will not stay at the stage of filling the gap of the project, but what we will expect that I attest my commitment as institution to be very active in achieving the goal of this Africa BioGenome Project by harnessing all the means that CNRST will have. So my presentation today will focus on implementing this strategy for biodiversity and how our institution, among others, will contribute to this implementation. So my presentation will have two parts. The first part, I will introduce my institution very briefly to be in a time. I will focus on the role of the functional genomic platform as a national core facility. And I will give you some information about this coordinated collection of Moroccan microorganisms as an example of how to preserve biodiversity at level of microbes. And the second part, if the time will allow me, will be focused on the key elements needed to implement a national biodiversity strategy and how my country, Morocco, comply with. So CNRST is a public institution in the framework of the law 8000 and has legal personality and financial autonomy. He has its own decision-making bodies, and it is overseen by the government. And also, CNRST operates under the Ministry of Higher Education, Research and Innovation. So my institution, the CNRST, is a key player in the national research and innovation system. And the main mission, if I could resume, is promoting, supporting, developing, and valorization of scientific research in Morocco. To reach these aims, we have some ambitious development strategy, and the last one is from 2023 to 2027. And to implement this strategy in order to support research, to facilitate research, to valorize research, we have these three main pillars as activity of my institution. It's not working. Okay. Well, it's working. Okay. So CNRST behave or act as a polling agency. It means this mutual use of all the tools that are needed for research. The second pillar is supporting research. I mean with that, financial support of research project, fellowship, mobility of researcher, and so on. And also the support, the valorization of the research. Given the time that is allowed for this presentation, I will focus only on the agency, the polling agency, which is a kind of mutualization of the main dedicated for research. So for CNRST, we have 5 divisions. The first one is EMIS, which means the Moroccan Institute of Scientific and Technological Research, which provide all the researchers across Morocco with this electronic access to scientific publication. And also we have some database. For example, all the theses are stored in the Tobqal database. This is the EMIS. The second one is ENG. This is the geophysics and the National Geophysics Institution, which is in charge of the seismic surveillance. And he has also a huge database that are very useful for the research. And the third one, and I belong to, is UATRS, which is mean technical supporting unit for scientific research. This is this core facility that provides services in terms of the analysis of all the university across Morocco. And this is very important. It means that researchers at any public university has a free access to our platform. They can perform all the analysis at chemical level, material science, and genomic also. CCMM, I will speak later on, and Marwan, which is this Moroccan-wide network that rely all the university to the internet and also host this high performance computing. So when it comes to speak about this core facility, it means how we can gather many scientific instrumentation to answer a question. So we have microscope, electronic microscope, scanning microscopic for material science and also for biological research. We have all this GCM, SMS, LCMS for chemical analysis. When it comes, for example, for valorization of medicinal and aromatic plants, we can perform chemical analysis, but we can also perform genetic analysis. And this is the way to make this connection between the phenotype or the chemotype and the genotype. And also, it is very important to have an idea that any living being cannot be reduced to only its genome. But functional genome, the transcriptome and proteome are very, very important when it comes to the adaptability. So supporting this technical support in Inite have, okay, I have seen already the mission. So to go, this is just to give you an idea about this infrastructure. 2018, we have this new building, and the image in the right here, this is the genomic functional platform, how our laboratory for DNA preparation, sequencing, Sanger sequencing, NGS sequencing, and how to prepare genetic material from any biological matrix. This is just to give you an idea, and the person who will perform her hands-on tomorrow will visit this facility. Just to give you an overview. So how this core facility will contribute to the biodiversity. So I have speak about all this chemical analysis that can be used for the valorization of natural resource, that can be applied to pharmaceutical, cosmetic, and so on. And the most important, what it comes in mind when we speak about biodiversity, immediately we speak about genomics. So we have more than 10 years experience with all biological matrix to prepare genetic material, to amplify the genetic material, to perform Sanger sequencing, to perform NGS sequencing, to perform genotyping, both by real-time PCR or by sequencing. And now I go to the Moroccan collection of microorganisms, which is very a concrete example how we can preserve and this biodiversity at level of microbe. So CCMM is located in University in Rabat. And the main mission is promoting investigation, preservation of national biodiversity in terms of microorganisms. And what we perform, well, it comes to go very fast from I don't know. Some change has been done when I sent my presentation. Yeah, I know that the original time is 30 minutes and it must be cut by half, but how to speed it? Okay, I will deal with that. So for the collection of microorganisms, what we done, we have all this capacity and this scale to isolate for storage, characterization. When I mean characterization and phenotypic characterization at level of microbiology, and also we perform 16S for the identification and whole genome sequencing for this bacteria. And we have this storage capacity, research unit and bioinformatics unit to deal with all genomic data. At the new facility of CCMM, we will have a new building in next year, inshallah, because this CCMM have been recently in 2018 designed as the first international depository authority, which means that when it comes to the valorization of microorganisms, I see lots of research dealing with the biodiversity, microbial biodiversity. And when it comes to identify this bacteria, how can we go to the valorization in different applications? It is very important to have this IDA, International Depository, because you want to have a patent that is related to this microorganism. And before 2080, there is no international authority depository at level of Africa. So we are really proud to have this. And I take this opportunity to invite my colleague from the country, from African country, that have very interesting bacteria that will be used for applied biotechnology. You have your African International Deposit Authority, and you can put your microorganism in. This is made by African for African, and it must stay in Africa. So this is an idea about our catalog. The second version is about 1,400 strain, and actually we have more than 2,000. I will go very quickly. We have some bacteria from environment, food, and medical. It is very important to perform all this analysis and biodiversity microbiome, but it is very important that the most important strain that will be used in agriculture or whatever, it must be storage for a long time in order to be valorized. So we have our online catalog, which is in English, and you can have all the information about the strain you will to retrieve. And this is an example of how this collection, how the research unit belonging to this collection explore the biodiversity across Morocco. We study different ecosystem, and we have different publication. I will go very, very quickly. We have isolated some specific strain, and we give them names of the region where they are isolated. For example, you have this Streptococcus tangeriensis. It means that it is isolated from Tangier. Yusufiensis from the Yusufia, we have retrieved and isolated from Moroccan phosphate mine. And CCMM is visible at global catalog. In 2018, I have already said that the WIPO attribute this statue of ELD to the CCMM. And we also contribute to the valorization of different strain with one of the most important achievement is this patent. We have isolated a bacteria that have a repellent activity against mosquitoes. And you can imagine what kind of application we can do with that. So the CCMM play an important role in preserving Morocco microbiological diversity. They assist Moroccan researchers in valorization their work and increasing the added value and their discovery by participating in Nagoya Protocol implementation and serving as Africa's first international depository authority. So I will switch to the next part of my presentation, which is not my specialty, but when I get this invitation for speaking, I say, I have to learn something else. We have to speak about biodiversity. I have done some research that I want to share with you. When it comes to biodiversity and to implement a national strategy wherever you are, we need this 12 elements in order to have this national strategy. And as it has been said by Professor Bouabid, that biodiversity worst scenario are waiting for the biodiversity in the future. And in order to fight against this, we must have our national biodiversity strategy. So I will go key by key elements. I will give the significance of each key and how Morocco deal with the implementation of these elements. The first key, what happened? It's coming? Shall I have 20 minutes. Yes. Otherwise, I will conclude. I go ahead. Okay. Well, this is the most I have deal with. Should I start with that and speaking about my facility, and I have done this gymnastic. 5 minutes. Okay, no problem. The first key element is establishing a task force or committee by the main stakeholder. What does it mean? It creates a dedicated task force for or committee composed of experts in biodiversity conservation. This group will oversee the development and execution of the strategy. I will go very fast. So the main stakeholder is the Ministry of Energetic Transition and Sustainable Development. And this is the focal point of the CBD in Morocco. And this set up a subcommittee for the biodiversity and to be in line with the Article 6, paragraph A of the CBD, which is the mandatory country must conduct first a national biodiversity study. It must develop its strategy, report, establish biodiversity information and information exchange center. This is what we call the clearinghouse mechanism on biodiversity, and Morocco set up this information exchange center, and you can go to the website and you have all the information about what Morocco have been done so far in terms of his national strategy for biodiversity and all protocol and workshop that are organized in this field. The second key element for implementing a national strategy is assessment and research. It means that we have to assess the country biodiversity and agriculture for native species, farming technique, environmental impact should be also inventoried, and invest in science, research, and data collection. For that, we have in this clearinghouse all the information about our national strategy and about national diversity. And also at level of CNRST, we have in 2040, we have organized the first workshop on how research and development comply with the biodiversity. The third point is policy development and stakeholder engagement. And the main idea behind this, it means that for biodiversity, we cannot only, the principal stakeholder will design this strategy, but we have different ministry and different departments that are implicated for that. So I will mention some of them. We have the Ministry of Higher Education, Ministry of Equipment and Water, and the Ministry of Agriculture via different, for example, the Ministry of Higher Education have set up what we call the National Agency for Medicinal and Aromatic Plant. And we have these two national plants, Maroc Vert and Green Generation. The main stakeholder is INRA, and our colleague from INRA will speak about. And also we have the Sherifian Office of Phosphate that are the main stakeholder in the policy development. The 5th point is the conservation of native biodiversity. We have to implement conservation program and protect area. And Morocco have already done that. We have some areas that are protected under the High Commission of Forest, Water, and Fight Against Desertification. We have our national park, and we have all the information that are in the clearinghouse. And I have to speak about the CCMA for the biodiversity, microbial biodiversity. INRA have his own research unit for the biodiversity. So I think that I will have not sufficient time to go through all these key elements. I will keep it for the discussion. Thank you very much.
Thank you so much, Dr. Fahim, for your presentation. So our next speaker is Dr. Yusr Hamdi. Are you there online? Dr. Yusr Hamdi, are you there?
Yes, I'm online. I was muted.
Okay, thank you. So Dr. Yusr Hamdi is going to talk about Accessible Pasteur Institute of Tunis capabilities for biodiversity genomics and bioinformatics. So Dr. Yusr Hamdi is a biologist and researcher at Institut Pasteur in Tunis, Tunisia. She is a specialist in cancer genomics and precision medicine. Dr. Hamdi is a member of several international consortia, including BCAC, CIMBA, and H3 Africa. She started in the field of human genetics with the Human Genome Project at Laval University in Canada. And after obtaining her PhD in 2009, is recently joined the World Health Organization and as an expert in genomics and member of the World Health Organization Technical Advisory Board in Genomics. She is now leading the Genome Tunisia Project and Per Medina Consortium to define the reference sequence of the Tunisian genome and to implement precision medicine in North Africa. Please join me to welcome Dr. Yusr Hamdi. The floor is yours. Thank you.
Yes, so thank you very much for the invitation. I don't know if you can hear me and if you can see my slides. Is it okay?
Yes.
Okay, I will just try to close the cam to be sure that we don't have any technical issues. So, so thank you again. So I'm going to talk about accessible capabilities for biodiversity genomics and bioinformatics at Institut Pasteur of Tunis. So I would like to thank my colleagues Qaiser Dira, Marwa Boujam'a, and Hisham Ben Hassin, who really helped me to prepare these slides. So Institut Pasteur of Tunis is one of the oldest institutions, research institutions in North Africa. It was founded in 1893 by Louis Pasteur and his nephew. And in few days, we will be celebrating the 130th anniversary of Institut Pasteur of Tunis. And then during the colonization, so it was led by renewed French scientists like Charles Nicolle, who got the Nobel Prize in Medicine in Institut Pasteur of Tunis for the work that he did there for 30 years and for his major discoveries on typhus, toxoplasmosis, and leishmaniasis. So after the independence, Institut Pasteur of Tunis participated in the eradication of several endemic diseases in Tunisia, such as malaria, and we also contributed to the national vaccination campaigns like polio and to the development of public health programs. And I have to say that Institut Pasteur of Tunis also significantly contributed to the control and management of the COVID-19 crisis. After 1995, Pasteur Tunis changed its status and became a public health institution with an administrative and a scientific board which reinforced its research and development activities. So in Pasteur Tunis, we have three main missions. So we are working on diagnostic, on health services, on research and training, and on bioproduction of sera and vaccines. And we are funded and led by two ministries, the Tunisian Ministry of Health and the Tunisian Ministry of Higher Education and Scientific Research. So for the diagnostics activities or the health services, we have 23 diagnostic labs that cover a full array of clinical and biological tests. And Pasteur Tunis is recognized for the expertise of some of his labs in some specialized medical biology fields. We are also considered as a national reference center for rabies, polio, bacteria, and also international reference center by the WHO EMRU region for polio and for HPV. Pasteur Tunis is also a center of vaccination for travels and a center for rabies treatments, and this is given for free to all Tunisian citizens. So for the research topics, so we are working on both communicable and non-communicable diseases, and we are applying genomics to improve and ensure a sustainable use of biodiversity. And as you can see, so for microorganisms, we are working on viruses, bacteria, fungi, parasites, and this is for a set of infectious diseases. We are also working on human diseases, including cancer, rare diseases, hematological, hearing loss diseases, aging, autoimmune diseases, metabolic disorders, and others. Here I'm showing the list of the research laboratory that we have at Institut Pasteur of Tunis. We have 10 research labs. And as you can see, I highlighted some of the keywords that shows that we are really covering almost all fields related to biology. So we have microbiology, biotech, immunobiology, epidemiology, pathology, genomics and oncogenetics, parasitology. And for data science, we have bioinformatics, we have biostatistics, biomathematics. We are working on theranostics, and also we have a specialized lab on clinical virology, which is a reference lab recognized by the WHO. In all these labs, in almost all these labs, we are applying genomics and molecular tests to really conduct the research that we are doing in Tunis. For the bioproduction activity, so we have the only bioproduction unit of vaccines and sera in Tunisia, and we are producing vaccines not only for Tunis, but also for different other African countries. And we have a big space in Pasteur Tunis that is really dedicated to the bioproduction of vaccines. And it's recognized at the international level because we are applying international standards. So the products that we are generating in this unit are BCG vaccines for TB and also BCG treatment for bladder cancer, but we are also producing therapeutic sera, anti-scorpions, anti-vipera, and anti-rabies. In Pasteur Tunis, we also have 4 specialized units. So the first one is called CVT2 for the valorization and technological transfer unit. And in this unit, so we try to use the research data that we are generating to get patents or to really produce products that are based on the research that we are doing in Institut Pasteur of Tunis. We also have a second specialized unit called PMO, which is a grant office that are helping researchers in scouting, in project supports, in fundraising, and also in scientific monitoring. The third unit called UNIS, for Unité Spécialisée Communication, Sciences et Société, and it's more for communication, dissemination, outreach, and it's more to bridge science and society. And finally, we have the IPT Omics platform, which is the technology platform that we have, where we have like sequencing platforms, and where we have biology, molecular biology equipment. In terms of human resources at Institut Pasteur of Tunis, we are more than 500 permanent position members, and this includes scientists, medical doctors, biologists, pharmacists, researchers, along with administrators, paramedics, workers, technicians, and engineers. Now, for the training activities. So Pasteur Tunis is affiliated to Université Tunis Manar, which is one of the best ranked universities in Africa. And we are like supervising graduate and postgraduate students. And we are organizing, I have to say that hundreds maybe of theoretical and practical courses in Pasteur Tunis. In terms of students, and I think that it's extremely important to talk about this because this is the next generation that will continue the work that we are doing. So in Tunis, in Institut Pasteur of Tunis, we are generating more than 400 students, and we are supervising them during the last 3 years. And this includes PhD, master, engineering, medical doctors, veterinary, pharmacy, and so on. And just to give you an example on the diploma that are supported during the last 3 years, it's more than 250 diplomas in all these fields. Now I'm going to talk about the institutional infrastructure in terms of bioinformatics, and I would like to thank my colleague Kaiser Dira, who is responsible of this infrastructure, who really contributed and succeeded to implement a high-performance computing cluster at the Institut Pasteur of Tunis to strengthen our capacity in omics and data analysis. And just to briefly define what we mean by an HPC cluster, so it consists of several compute servers that are networked together, and each server is called a node, and the nodes in each cluster work in parallel with each other, boosting processing speed to deliver high-performance computing in our institute. So here the HPC infrastructure. So we have the computing infrastructure and the storage infrastructure. The computing one includes a master or a head node with 5 computing nodes and one high memory node for high demanding memory jobs. And for storage, we have a storage server with 96 terabits of capacity and a backup server with the same capacity. So here I'm showing the infrastructure, how it's really connected. And I have to say that all this infrastructure that is common for all those who are working in Pasteur Tunis is funded by the Tunisian Ministry of Higher Education. So I'm showing here the layers of our cluster in Institut Pasteur of Tunis. And here we have the nodes, the servers that are configured by Bright Cluster Manager, as you can see here. And the bioinformatics jobs are managed by the Slurm scheduler. And all this form the high performance computing cluster that we have. In terms of capacity, so we have for the CPUs, we have 272 cores. For the graphic processing units, we have 128 gigabytes. For the RAM, around 750 gigabytes. And for the storage, as I said, 96 terabytes in the main server and the same capacity in a backup server. And according to the LIMPAC benchmark, IPT, the HPC that we have, achieves a peak performance of 8.7 trillion double precision floating point operation per second, which is really amazing. So now, how we can access to this cluster. So it's available and it's accessible to all those who are working in Institut Pasteur of Tunis, but we have documentations, guidelines, and policies that we use to really get access to the infrastructure of bioinformatics. And this is like a kind of a request form that should be filled. And then you will have an account with a username, a password, and you can then use this platform. And it's exclusively to our workers and researchers from Pasteur Tunis. The data that we are generating and that we are storing is under the control of our institutional bioethics committee and the National Authority for Protection of Personal Data. that both are organizing several training activities on bioethics and research in the medical field, along with a regional training center led by the WHO and based in Institut Pasteur of Tunis. Several other training activities on how to use the infrastructure of bioinformatics that we have that is dedicated to the researchers, but also we are organizing specialized training activity for project leaders to know how to make use of the infrastructure that we have. So we are also a member of the H3 Bionet consortium, and we organized several courses in Institut Pasteur of Tunis, and my colleague Melik Shawsh is really leading this activity. So for data analysis, IBT courses has been organized for 3 or 4 years, and NGS Bioinformatics as well for 3 years successively. And lots of other training activities has been organized in Tunis, mainly the African Genomics Medicine Training course, and also the development of the African Precision Medicine Portal. In the framework of PerMedina project, which is a project on personalized medicine implementation, we organize the hands-on training using the HPC capacity in the framework of this project, and this is mainly for RNA-seq data analysis. And in this event, we invited researchers and young researchers from Morocco, from Algeria, and from Tunisia to benefit from this training. So now the last part of my talk will be on the wet lab infrastructure that we have. So for biobanking, we have a quite nice biobanking infrastructure with more than 100 equipment, freezers, refrigerators, liquid nitrogen containers. For nucleic acid extraction, we are no longer using kites because we have a very high throughput. So we are using robots and automated machines in the Institut Pasteur of Tunis. And the material that we are extracting, we are really doing the quality control using different machines, as you can see, to get DNA, RNA, proteins, microRNAs, and all biological material that we can extract. And for PCR infrastructure, we have more than 63 thermocyclers, light cyclers, and all the equipments that are needed for PCR amplification. For the sequencing infrastructure in Institut Pasteur of Tunis, we have the 3 generation of sequencing. So we have 3 sequencers from Sanger sequencers. We have 3 NGS machines, and NextSeq, and iScan, and NextSeq, and we have 3 MinION from the Oxford Nanopore Technology for long reads. So all this infrastructure that we have and the personnel that we have, we are participating in different research projects, as you can see here. So in 2022, we are at more than 100, this slide, I think that's not updated. So we are at more than 100 projects that are running in Institut Pasteur of Tunis. And we are funded locally by the Institute. We are funded nationally by the 2 ministries of health and higher education. And we also have international funds. And the total for 2022 is more than $30 million. The main funding agencies are in this slide. So the European Commission, the NIH, and the Wellcome Trust, mainly with the WHO and the Institut Pasteur Network. And for sure, the Tunisian government is really contributing in funding our jobs. So in terms of publications, so we are doing well. And during the last 10 years, we are at more than 150 papers per year. And recently we reached more than 4,000 citations for the publications of researchers of Institut Pasteur of Tunis. Here, very briefly, I'm showing the countries with whom we are collaborating in our research project. So we are covering almost all continents, but the main ones are France, USA, South Africa, Algeria, Bahrain, Saudi Arabia, and for sure, Morocco. Finally, I cannot talk about Institut Pasteur of Tunis without talking about Pasteur Network. So we belong to this network, and I have to say that we are the first Institut Pasteur that has been founded after Pasteur Paris. And so we are collaborating with almost all these Pasteur institutes all over the world, mainly those who are in the region, Algeria, Morocco, and Paris, but also with Conakry, Dakar, and other Institut Pasteurs in the world. So that's it. Again, I would like to thank the organizers for the invitation. I would like to thank you for your attention. And I think that in this talk, I've been able to show you a bit the capacities that we have at our institute. We are using this capacity for the works that we are doing on biogenomes, on humans, on animals, on microorganisms, and for the agriculture. And we are facing several challenges. It's the case in different limited resources countries, but we are trying to overcoming them. Thank you very much.
Thank you so much, Dr. Ramri, for Hamdi, for your presentation. We take questions for the two speakers. And we will just do the other presentation after lunch. So, do you have any questions? Online participants? Yeah. We have a question from Mr. Oluwasegun Babailei. Please unmute yourself and ask your question.
Hello, good afternoon. Hello, can you hear me?
Yes.
Yes, good afternoon. Please, I want to ask a question from the second presenter. Please, I want to ask that, can you show the slide where you showed the link to the bioinformatics in your institute? And also, I want to ask again that, how can you help somebody that is not from your country to come to your country, as in to do 1 or 2 research work in your lab over there?
Yeah, so thank you very much. In Pasteur Tunis, so the capacities in bioinformatics that we put in place is mainly for researchers and scientists working at Pasteur of Tunis. But we are collaborating, as I said, with different other groups in Africa, different other research teams. And I have to say that we have lots of African researchers and young researchers that are working at Institut Pasteur of Tunis from the region, from Mauritania, Morocco, Algeria, Libya. They are working with us, but also we are training different other young researchers from Mali, from Senegal, mainly from Francophone countries, I have to say, because they are more familiar with the language. But I don't think that we have any issue to get people involved in the institute. So I can keep my email address on the chat box, and if you are interested to collaborate, please just drop me an email and we can discuss.
Okay, thank you. Other questions?
Question to Professor Fahim. Direct question. Have you any plan on how to combine the National Strategy of Biodiversity with the AfricaBP project? That will be good to answer for us and the people that are online. Thank you.
Yeah, for sure. I have already seen that we are really delighted to be a partner of this biogenome project. And the question, how are we going to contribute to achieve the main goal of the Africa BioGenome Project? So I don't have time to show you, but there is very important information about MyUnity, which is the functional genomic platform. Recently, we became a collaborating center of EIA, the International Atomic Energy Agency, which provide a lot of financial support and the training in different fields. When it comes to speak about EIA, the first thing that comes in mind is the peaceful use of atomic energy. But there is another very huge activity related to human health, to animal health, and also to agriculture. And I advise all the researchers also to go to this website and to take over all the information about the support that are provided by this EIA. Since we become this collaborating center, it means that we will have access to all the capacity of EIA and all the laboratory. So we are going to harness this accessibility in order to develop all this big project on genomics, because it is very important to have high capacity for dealing with this data. We are going to generate a lot of genomic data, where we are going to storage this, how to deal with. All these things will be arranged by this in the framework of this collaborating center with EIA.
Okay. Other questions? No. Okay, we stop then this session for the lunch break. Meet you all in 1 hour. So, thank you. S'il vous plaît, installez-vous, qu'on puisse commencer. S'il vous plaît, on va commencer bientôt. Ça fait? Okay, we are set to start again. So we will be finishing the session about regional biodiversity infrastructure capabilities with our next speaker, who is Dr. Rashid Mentag. He is a distinguished plant biotechnologist holding a PhD in plant biotechnology and an MBA in bioindustry from Canada. He has been a dedicated member of INRA Morocco since 2006 and currently serves as a researcher, director de recherche at the biotechnology unit of the Regional Center of Agricultural Research in Rabat. Dr. Mentag's research portfolio includes recent contributions to genomics, bioinformatics, and plant pathogen interactions. His active engagement extends to numerous research projects where he assumes role as the principal investigator for both national and international initiatives. Additionally, Dr. Mentag has made significant advancements, holding 3 patents related to biocontrol and tree micropropagation. Beyond his research activities, Dr. Mentag is a committed educator, actively participating as a member of teaching teams at Moroccan universities and institutions. Please join me in welcoming Dr. Rashid Mentag as he shares his expertise with us today.
Assalamu alaikum. Thank you. It's a pleasure to be here. First of all, I would like to thank all the organizers for this nice and interesting event. Through this presentation, I will talk on some landmarks, genomic landmarks of date palm at national and international level. But before that, I will let me begin with understanding the fundamental concept that has shaped agricultural history and continues to influence our modern world. This fundamental concept is a process, is the cornerstone of the human civilization that entertain a relationship between human and other kingdom like plants and animals. This concept is domestication. So what is domestication? Is the process by which a wild plant is changed through artificial selection to better fit human needs. So this process started 12,000 years ago and still in process through a co-evolution process. And there is about 2,500 species that are semi or fully domesticated. And these crops are major crops for humankind. And why it's interesting to understand domestication? Because there is a lot of genetic mechanisms underlying domestication and diversification of cultivated species to gain insight into the genetic basis of traits for crop improvements. So we want to understand this genetic basis to use them for improving genetic breeding of plants for improvement. And mainly only annual crops have been fully studied in this kind of domestication. And there is a lack of information about the perennial crops. Among these perennial crops, there is a fruit tree species, and date palm is among these species. So we have now some evidence that date palms started cultivation since 7,000 years ago. And we know that the natural geographic distribution is among the MENA region. It means North Africa and Middle East area. But there is also some countries, other countries such as Pakistan, China, and US that are producer also. So all over the world, there is about 150 million palm trees. And among the Arabic country, there is about 100 million trees of date palm. And also we counted about 5,000 cultivars or varieties of date palm in 30 countries. In Morocco, we have about 453 cultivars of date palms. Among these cultivars, you may know Majhoul, the famous Moroccan variety. So why date palm genomics? The importance of this crop has generated significant interest in the genetic basis of its traits. For example, we use it for increased production. We use it also for the quality of the fruits and also for resistance to biotic and abiotic stresses. However, there is some structural limitation or constraint to the development of classical genetics. I mean by that, the mapping populations, like for annual crops, we can do crosses, but with date palm, it's really time consuming to do so. So we have to manage these constraints. Among these constraints, so we have we have the cycle development of the date palm. So it takes 4 to 6 years to the first flowering of the plant. And also there are, if we want to generate offshoot of vegetative propagation, the offshoots are this element here. So to do multiplication of this individual, so you have to wait until the production of this offshoots. And this offshoots, there are not many offshoots produced by one tree. For example, for Majhul, we have all the life cycle of Majhul. He can produce only 20 offshoots. So it's difficult to manage. And also for the well characterization of the tree, we have to wait 10 to 15 years to have the maximum yield. So all these constraints don't allow us to use mapping population. So this is why we should use genomics to understand and to identify some interesting genes and QTL that are associated with interesting traits. So for there is a lot of publication on date palm. There are about, until 23, there are 3,600 publication. Among this publication, there are only 79 papers that present genomic studies. And if you see here, 2009, for example, at the NCBI gene bank, we had only 100 kilobase per date palm DNA sequence available in the NCBI. And starting from this date, we get increasing data of sequences in date palm. And the first effort on genomics have been applied to organelle of date palm. So we have chloroplast genome that have been sequenced in 2010, and we have also mitochondria that have been sequenced in 2012. Why it's interesting to have this kind of data? Because for several, to respond to several questions. For example, with this data, we had been able to verify the real taxonomy of date palm, because date palm, when we are doing a phenotyping characterization before the venue of the genomics, we thought that we have more than 20 species belong to Phoenix genus. But after sequencing, we got less species, and we confirmed that there is only 13 species belong to Phoenix genus. And also the mitochondria and chloroplast, there is some interesting genes that are involved in photosynthesis and in drought resistance. So it's very interesting to know this and to characterize these genes. Now, the question is, how about the genome size of the date palm? From the beginning of the research on genomics of date palm, there is many studies referring that there is about 700 million base to 1,400 million base. But recent study conclude that the real genome size is about 900 million base. So starting from that, we can check the first attempt to the sequencing of date palms. So it was a Qatari team that succeeded on the first draft genome of the date palms. They were able to assemble about 40% of the genome. After that, a second attempt in 2013, they succeed. Saudi Arabian teams succeeded on having about 60%. After that, and all the previous sequencing on female plants, because you know that date palm is dioic species. So we have male and you have female tree. And in 2019, the first time the Emirati teams succeeded on assembling about 85% of the genome, the date palm genome. It was a male. And the last one is female Khalas cultivar, and it's almost the complete genome. But you can see here that there is 900 scaffolds. So it's still not complete and high. It's chromosome scale for sure, but it needs more improvement. So here is the map of international collaboration in date palm collaboration. So you can see the major acting teams are from Saudi Arabia, Emirates, Qatar, and also China and US. Now, how about transcriptomics? We know that transcriptomics allow to identify differentially expressed gene pathways and regulatory networks associated with specific biological process in plants in general. So the work in transcriptomics there are no many studies on transcriptomics. There are about 14 articles focusing on transcriptomics. And these articles mainly focus on genes involved in fruit color, salt tolerance, resistance, and sugar metabolism. How about genotyping in date palm? We know that genotyping is the discipline that aims to detect genetic variation at the DNA level, at a specific position across all or a portion of the genome. For that, we use molecular markers and in sometimes sequencing, like genotyping by sequencing. So molecular markers allow us to understand many processes, but specifically, we can use them to identify the polymorphism among species, among varieties, or among individuals. So here is some work that has been done in transcriptomics. Few studies, 18, mainly this distinguishing between different cultivar and sex determination in date palm. So this is the major use of genotyping. Another interesting topic is genome-wide association studies. As we could not develop mapping population in date palm. It have been done in the past in 1945 in US, developing some, improving some males, date palm males, but in general, we don't proceed on mapping population. So we use GWAS. For that, we know that GWAS are a powerful method for mapping the association between genetic variation and phenotype. And after that, we can identify a candidate genes and genetic markers associated with important agronomic traits. Here we can see some results on the GWAS applied for 6 determination region in the date palm. And we could see here that there is some high spot for in the chromosome 18 for 6 determination. And also GWAS have been used for fresh fruit color in date palm. And here also we can see a hot spot in the chromosome number 4, and also for dry date palm fruit color. For sure, we will have a lot of information coming from genotyping. Lot of databases. I will go through this quickly. So we have measured these 2 databases that refer to some interesting data coming from genotyping and sequencing. For date palm genomics at INRA, we started with the adoption of sequencing protocol Ion Torrent and also Illumina Next 550. And we have this kind of results coming from the 2 platforms. And also we proceed with the sequencing of Majhoul, Boufkous, Bouskli, Nejda, Aziza, Bousthammi, and 2 males. Bioinformatics, I will go quickly. Bioinformatics analysis allow us to identify some interesting SNPs and indels, and like we see here, for example, and that are that can distinguish among the species. And also we proceed with the analysis of about 20,000 samples for the Ministry of Agriculture. Okay, so here we know that that in 2017 we know that there is a 2 spot of diversity of date palm. The main one is located in Middle East, but we know that there is a population that are more diverse in the North Africa. And we don't understand what happens and how it's, if this population comes from Middle East, how could we explain why it's more diverse than the original one? So for that, sequencing work has been done, and we understand now that the Middle East population, through migration, have been crossed with Phoenix through Theophrasty. I always forget the name. Theophrasty from south of Greece, and this crossing give more diversity from the original population coming from the Middle East. And also through genomics, we know now that for 6 determination, there is evidence that the date palm is XY system. And Morocco possesses a large date palm genetic resources, and efforts to characterize and preserve these resources are necessary. So as a conclusion, we can say that the study of crop origin is of great interest, both in field of evolutionary biology, because we understand, we confirm that this species belongs to this genus, but also we understand also that there is some interesting gene pools that can be used to plant breeding. But still, there is some question marks belong to the domestication of date palm. And we know also that there is a wide relative species named Phoenix Theophrasty, has contributed to the diversification of date palm, specifically in the North Africa. And also I hope that we will use our genetic resources and we will work all together on characterization of our rich genetic resources, especially date palm resources. At the end, I would like to thank all these nice people from INRA working on different regional centers for their contribution to genomics and genotyping of date palm resources. And thank you very much.
Thank you, Professor Multak. Thank you. So since this is the last presentation for this session, I open the floor for questions. Stand up, please.
Hello, everyone, and thank you, Dr. Multak, for this insightful presentation. So, as you explained, like the cycle of the palm trees is very long, and I would like to ask about the strategy of breeding for these palm trees. Like you explained the ways to identify the genes that are related for like traits like yield. So I would like to know how we can use this information to shorten the cycle for breeding. Thank you, sir.
You are welcome. So here in Morocco, we mainly use, I don't know how to say that in English, but sélection massale. Selection massale, I don't know what we can call that in
mass selection.
So it means that we go to the field and then we see the behavior of the individual, and then we can compare with the others, and then we can choose depending on the characteristics of these individuals, and then proceed for the in vitro, for example, multiplication to have multiple copies, and then it works like that. For example, for the development of Nejda cultivars, there is the strategy we used for the selection. But what is interesting with genomics, for example, if we know that there is some interesting traits for resistance against some pathogen, for example, for biota, still right now we don't have this information. But for example, in the future, if we linked the resistance to biota to one specific allele, for example, so we can do PCR or a real-time PCR and check some individuals and do selection based on the molecular selection. So this is a strategy to be used and to shorten the development of development of new cultivar or new varieties. It has been done for the development of new male in California. The program started in 1945 and ended about 1980. And you know, in such exercise, you have to start the crossing now, and then another colleague will continue with your population. So it's not easy with the perennial trees, but it's interesting. If we have long-term strategy, it could be interesting also.
Sorry again, can I ask another question, please?
Yes.
So, like, if we have this gene of resistance for like this disease, we can't integrate this gene in the like Majhul variety if we want to incorporate this gene, or we need to have a long way
Yeah, there is a limitation on proceeding on the integration of external genes, because we are respecting some administrative rules that there is no GMOs, but still there is a new strategy like genome editing that could be used, but this is also a public concern, and some ones say that is GMO, the others say no, this is GMO, but forget about genetic engineering. We can use markers to select a specific individuals that have some interesting traits, different traits, quality, resistance to pathogen, or resistance to drought. That could be selected among, because we have a huge genetic resources diversity in Moroccan mangrove of date palm in Morocco. So we have to explore this interesting genetic resources.
Thank you, sir.
Any other question? Have question?
Bonsoir tout le monde. Alors, est-ce que je peux
Parler
poser
en
une
français.
question en français?
Allez-y.
Seulement, vous avez parlé de fusariose, de fusarium. Alors, je veux savoir seulement si on possède, on dispose au Maroc des techniques de purification de l'ADN déjà extrait du sol sur le fusarium, du champignon de fusarium. Parce que j'ai des connaissances sur que les laboratoires des analyses agroalimentaires au Maroc ne disposent pas, je parle sur 2019, des techniques de purification. On dispose des experts d'identification de fusarium, des techniques d'extraction, mais toujours on est en dépendance avec des laboratoires à l'extérieur pour faire la purification. Et quels sont les progrès qu'on a réalisés dans l'étude de cette maladie de palmidati? Merci.
Oui, donc par rapport, est-ce que vous voulez que je réponde en français ou en anglais?
Oui.
Par rapport au travail qui a été réalisé sur le séquençage, notre équipe donc, a fait jusqu'à date 2 publications. Il y a une publication qui va sortir dans les jours qui viennent, c'est accepté. Sur les 2 premières, c'était des draft genomes du fusarium oxysporum forma specialis albidinis, qui est spécifique au palmidati. La 3e publication, c'est une publication beaucoup plus approfondie et il relate un petit peu le chromosome scale, c'est-à-dire assemblage au niveau chromosome du fusarium oxysporum albidinis. D'accord? Et donc, on a pu déterminer qu'au niveau du fusarium, on va l'appeler FOA, fusarium oxysporum albidinis. Donc, au niveau du FOA, il y a des régions qui sont des régions des chromosomes qui ne sont pas uniques nécessairement au FOA, mais qui sont présents aussi chez les espèces de fusarium oxysporum. Parce qu'il y en a à peu près 160 formats spécialistes qui est spécifique à chaque plante, à chaque culture, comme la tomate, comme le radis, ainsi de suite. Donc, chez le FOA, on a identifié des régions qui sont, on les appelle les lignages spécifiques. Ce sont des chromosomes qui sont responsables de la pathogénicité. Donc, maintenant, si on veut faire un dépistage au niveau du sol pour identifier le fusarium, on peut faire un dépistage en utilisant des amorces qui sont prises au niveau des chromosomes qui ne sont pas nécessairement uniques. Mais dans ce cas, si on fait un dépistage, ça peut être positif alors qu'en fait, c'est un fusarium autre que FOA. on peut avoir ce genre de résultats. Mais avec cette ressource génomique qu'on vient de publier, il va devenir beaucoup plus facile à développer des amorces qui sont spécifiques au FOA et qui pourront être utilisées pour déterminer est-ce qu'au niveau d'un sol particulier, est-ce qu'il y a présence, oui ou non, de Fusarium oxysporum albidines, la forma, c'est-à-dire la forme agressive, parce qu'il y a des Fusarium oxysporum albidines qui ne sont pas agressives. Et donc, c'est beaucoup plus spécifique pour identifier les agents vraiment virulents.
Oui, merci beaucoup, professeur Milta. Merci à vous, merci à l'audience. Donc, we close this session. And before opening the next and last session for today, I just want to check if Dr. Thank God Ebenezer is online.
J'ai plus la main.
Yes, I'm online.
Okay, perfect.
J'ai plus la main. Est-ce que je peux juste lancer la main?
Oui.
J'ai plus la main. C'est bon.
Okay, let me introduce Dr. Ebenezer, who is a bioinformatician based in the University of Cambridge, UK, where his work is instrumental in developing computational pipelines for early cancer detection and prediction. Today, Dr. Ebenezer joins us to shed light on a critical initiative. Apart from his significant contribution to dry lab science, he has been actively involved in fostering collaborations, both on the international and regional stages, and at the local level, sorry. His recent involvement in the establishment of the African BioGenome Project, AfricaBP, carries immense importance. In this session, we will delve into the proposed AfricaBP Open Institute Fellowship in Biodiversity Genomics. Dr. Ebenezer, the floor is yours.
Thank you so much. I have one of my colleagues here, Professor Julian Osuji, who will do the presentation. Professor Julian, are you on the call today, please?
Yes, I am.
Okay.
Thank you. Good evening, everyone.
Okay.
Yeah, I guess I'll be allowed to share my screen.
Yes, please. Please go ahead.
Yeah. Can everyone see my screen clearly?
Yes, please. Carry on.
All right. Thanks so much. Yeah. So the AfricaBP Open Institute Fellowship Program in Biodiversity Genomics is a way of AfricaBP trying to drive the import of the program home. We all know that Africa is rich in biodiversity, which is the genetic bedrock for building sustainable and climate resilient ecosystems. This biodiversity supplies us food, promotes good health, offers alternate and clean sources of energy, offers the impact of climate change, and supports livelihood. Unfortunately, this biodiversity is under threat. Very serious increasing threat, which has led to loss of biodiversity, which has far-reaching and damaging effects on all life forms, our environment, and livelihood. To address this, to react to this, Africa BioGenome Project actually has 5 grand challenges, and then one of these grand challenges, technology development, knowledge exchange, and industrial grand challenge, offers us a way out. And through the knowledge exchange aspect of this grand challenge, AfricaBP established the Open Institute for Genomics and Bioinformatics. This Open Institute is a capacity building interface for Africa BioGenome Project. And does this capacity building in 2 ways, through workshops in 2 layers. The first layer being through the straight from the Open Institute direct And then the next one is the two regional hybrid workshops, such as the one we are participating in today. And then in these workshops, the Open Institute also tries to open, that's the next approach is to establish graduate fellowship programs. And what gave this initiative a bite was what happened in the first regional hybrid fellowship, workshop, which was hosted by the University of Port Harcourt in Nigeria, where a World Bank funded Africa Center of Excellence offered 20 scholarships for 20 prospective Africa BioGenome Project members in the area of biodiversity genomics. That graduate fellowship is an international exchange fellowship. And we hope, and it's growing as we stand. Now, the fellowship has different research teams. We have the sampling and biobanking, in which deals with the processes and SOPs involved in sampling and then biobanking in order to preserve species in Africa. We also have the Biodiversity Genomics, which aspires to look at the genomics of mangrove system, that's plants and non-agricultural animals in the brackish ecosystem. It also aspires to look at genomes of livestock, plants, medicinal species, raw materials, industrial raw materials, and so, etc. And then also aspires to look into the ethics, policy, and social issues that surround this genomic initiative. This fellowship program actually has two interest groups. One is the Africa BioGenome Project on one side. The other is the Africa Center of Excellence, field Center of Excellence on the other side. The key Center of Excellence is the Center of Excellence in field research, AC4. And then we also have the Public Health and Toxicological Research, which is also World Bank funded, which has recently shown interest to support the ACC4 in this initiative. They have this interest in promoting homegrown research networks for human capital development, especially through partnerships and network of higher institutions. They have funding for home tuition and some of the expenses in Nigeria for at least 20 prospective PhD students. They recognize the capacity of AfricaBP for assessing regional potentials for biodiversity genomics. And then because of that, they called for this scholarship program. Well, the program actually aspires to generate sequence data that contribute to knowledge and technology transfer, provide support for bioinformatics and genomics capabilities, and leverage on already established partnerships. The objectives, which are actually derived from the research teams, include biodiversity genomics, and then tries to cover agricultural, medicinal, industrial raw materials and genomics, and then ethics, policy, and social issues. Then the sampling and biobanking. The idea is to produce at least 20 PhDs in 3 years. In this arrangement, AC4, ACC4, and then possibly supported by the Pluto, will take care of the home tuition fees, accommodation costs, research, and living expenses of the individuals in this fellowship program. And then AfricaBP partner entities will cover the cost in countries of affiliation, research, accommodation, and living expenses for the period they stay away from Nigeria. Basically, basically the funding structure is what we've just seen. And then we have at least 20 PhD graduates after 3 years, at least 2 reputable journal publications per candidate at the end of the program, sport capacity building and academic mentorship, and more biodiversity data for agri-conservation and biodiversity genomics in Africa. Thank you for listening.
Yeah, thank you, Professor Julian. Sorry, moderator, can I add a few comments? Professor Julian, do you want to just stop sharing, please?
Okay.
Yeah, so just to add to what Professor Julian has said, when we started AfricaBP in 2021, when we launched the program, one of the things that became clear is because our main aim is to sequence the genome on the ground in Africa. And we all know that Africa has challenges, when it comes to infrastructure and capacity compared to North America and Europe. And to be able to do that, the main thing we've focused on is how do we sequence the genome on the ground in Africa, and how do we work with African institutions, for instance, like UM5 and also UMP6 and all the whole African institutions. And in this case, the institutions in North Africa. How do we work with them to sequence these genomes? So we came up with basically there was a knowledge exchange program or grant challenge, which actually midwifed this particular workshop. So, and this particular workshop is just 2 of the 5 steps we are taking particularly. So the day 1 and day 2 of this workshop focuses on awareness creation. So that is where we come together and then we try to understand the current understanding of biodiversity genomics across Africa. Then the day 3 to 5 focuses on the capacity building in terms of very short, intensive capacity building efforts for 3 days. And this will start from Wednesday until Friday. Then down the line, we again have some other approaches in the pipeline. And one of those is the fellowship program. So the fellowship program, for instance, we are looking at the PhD fellowship programs, and are also looking at group leader fellowship program or group leader positions, as well as so many others within the Open Institute. Because the reason for doing this is that we want African people to sequence this genome themselves. We want institutions to connect to each other and to, within Africa, across country, with these countries, to actually do this sequencing particularly. So the first workshop, regional workshop of the AfricaBP was held in West Africa. And just as Professor Julian mentioned, there were interest to actually, do this. And one of the things we are looking at is the PhD students will be affiliated with the institution. And just to mention that this presentation that Professor Julian just made is not specific to the University of Port Harcourt. So if, for instance, at the end of this meeting, there are partners and sponsors who actually want to do this for North Africa, please get involved with Professor Boabid, because we will be connecting this to the regional levels and then bringing it at the top at the continental level. For instance, if there are partners that are willing to actually support and fund, for instance, a PhD position or postdoctoral position or other aspects to actually build the capacity, particularly. And then we moved from West Africa, Nigeria, and then we went to South Africa in August, and then we organized there. In South Africa, we had around 1,200 registered participants, and now we're here in Rabat, Morocco. And after this meeting this week, in December, we'll be going to Nairobi, Kenya, where we also have over 1,000 registered participants. And then the cycle will begin again in Port Harcourt, Nigeria. So this is just more of an overview, and I will be happy to actually answer any question and kind of also have a chat and discuss about this. Thank you.
Thank you so much, Dr. Ebenezer, and we are grateful for the initiation of this movement and for all the work you are putting to enhance the African science and improve the African capability building. So is there any question for the speakers? Okay, there's one here.
Thank you. Thank you, Prof. Julian. I have just a question about the expected outcomes. You said that, just a comment, not a question. More data about genomic resources in Africa will be generated. I'm suggesting to be more specific on this issue because we are looking for sequencing that genome species on Africa. So if we have 20 PhD scholarships, let's say that we need to generate 20 genome sequence from African species. Thank you.
Do you expect the question and the answer immediately or later?
Maybe I think you can go ahead, Professor Julian, and then I can support your answer.
Yeah, I think I didn't get all the question clearly, but I know he was talking about specificity in the program, like 20 people, for instance, coming to take up PhDs. The PhDs should actually be in the area covered by the interest and mandate of Africa BioGenome Project, genomics, sequencing of species. In this 20 PhD, someone possibly might be interested in sequencing the genome of clarias in a particular environmental location. And another person might be interested in sequencing the genome of Clapatonia in another environment. But then they're all programs designed with the same structure, issue the certification with the same certification and all of that. And the program so supervised accordingly based on the interest of the candidate. So it's not a blanket thing of any person just does whatever is stuffed into the mouth. Each person has a proposal so far as it flows with the interest, the line of action and the operation of interest of Africa BioGenome Project, genome sequencing of non-human species that agree with the mandate of the Center of Excellence or the program that anchors the PhD work. I don't know if I'm clear enough.
Yes. So, Prof, let me just support your statement. As I mentioned previously, Slamin, is that the PhD position that Professor Julian is talking about, the 20, is just emanating through one of the AfricaBP regional workshops, and the University of Port Harcourt has made interest and shown interest to sponsor 20 PhD students in biodiversity genomics. So if University of Port Harcourt make commitment in sponsoring 20 PhD students, then another university in North Africa or in Morocco, we have 1 or 2 or 3 or 5 or 10 universities making such commitment in sponsoring 20 or 10 PhD students. Then at the end, we'll have 100 PhD students across Africa. If 100 PhD students across Africa are working in biodiversity genomics, then one PhD student is tackling 4 species. The reason why I say 4 species is that recently we published in the paper Nature Biotech, whereby we estimated that if we have one PhD student or fellow actually working on 4 species, then in 10 years' time, we'll be able to build 200,000 capacity. We're able to build the capacity of 200,000 African scientists, thereby closing the gap in genomics across the continent. So I want us to see this as not just a University of Port Harcourt activity in terms of the PhD sponsorship. It just happens to be that we held the workshop in Port Harcourt at the first time, and then there were interest from the university to sponsor PhD students in that. So we're also hoping that more African universities can actually show interest and come on board and sponsor PhD students in their locations, in their universities. It could be PhD, it could be postdoc, it could be early career fellowship. So the main idea is that we believe, and I believe, that if each university is in each country, at least one university in one country, and I do know that there are more than one universities in each African country, but let's assume one university in one African countries, then that would be more than 20 PhD students who will be working on this, whether postdoc or researchers. And ultimately, that will build up, or it will have direct impact or direct correlation to the number of data that will be generated for the countries involved at the national level for the use of the people at that level. So I don't know, Slemi, if that answers your question, please.
Yes. Yes, thank you. Other questions? Okay, if not, so just thank Dr. Ebenezer and Dr. Osuju for their presentation. Thank you.
Thank you.
Thanks. You're welcome.
So close this session and we move to the practical exercise introduction, section 2, which should be presented by Professor Bouabid Badawi. I don't see here. Okay. Either the second speaker is not here, Mustafa Fahim. So we take a 4-minute break since we try to reach to Professor Badawi, and then we start again. Okay. Okay, our first speaker will be Dr. Slimane Khayi, who earned his PhD from the University of Paris-Saclay. Following that, he served as a postdoctoral researcher at the Institute of Integrative Biology of the Cell in the south of Paris at Gif-sur-Yvette, specializing in the study of soft-rot bacterial pathogens, focusing on several aspects such as genome assembly, evolution, and population genomics. Subsequently, Dr. Khayi transitioned to INRA in 2017, where he assumed the roles of bioinformatician and genomics expert. At INRA, he dedicated his expertise to crafting high-quality genomics toolboxes aimed at addressing challenges associated with the development of national agriculture. Dr. Khayi is serving in African BioGenome Project as co-chair of the Sequencing and Assembly Subcommittee. His contribution to this remarkable initiative include efforts in capacity building and the assembly of sequenced African species. Sorry. So the floor is yours.
Thank you. First of all, I would like to thank Professor Boabid Badawi. He is not here for this insightful workshop. Okay, thank you for this insightful workshop. And also many thanks are due to the organizing committee for huge efforts deployed for the success of this workshop. At INRA, we are honored to be a partner of African BioGenome Project. This fantastic initiative in which we trust at INRA. And we believe that this project will solve several issues in the continent in the long term, such as food security and the erosion of genomic resource on the continent. Also, we are honored to partner with AfricaBP and to host this practical workshop in which we will deliver some hands-on genomic sequencing and bioinformatics. We are believing that this workshop will contribute to the capacity building in well, we believe that this workshop will contribute to the capacity building in biodiversity genomics on the continent. I will be very brief. I will give you at a glance what we will do at INRA for the coming 3 days. So can you see my slide? No. Yeah. So I will give an overview of what we will offer to the participants in the coming next 3 days. So the workshop will be focusing on mastering Next-Tech 550 Illumina sequencing machine, combined with bioinformatic analysis. Before delving into the content of the workshops and logistics, so the workshop will start tomorrow. We'll start from 8:30 to 5 p.m. Using time zone GMT +1. So the workshop will be held on the Regional Center of Laboratory Research at INRA, located at the Avenue of d'Espagne, and the location was sent already to the participants. Of course, we are offering transport to the participants upon their request.
and all the logistics necessary to that is established. So the workshop will be organized with my colleagues from our unit, Dr. Rashid, me and Dr. Rashid Mintag, Dr. Bisaraki, and Mr. Amin Al-Muhtadi. So a quick introduction about INRA for to know who is the institute. INRA is National Institute of Agriculture Research. INRA is the French name. We are organized in 10 regional centers that try to cover all agri-systems around the country. We have 10 R&D services, and we have 21 experimental stations across the country, as I said, representing all agri-systems. We are 30 research units distributed across the 10 regional centers. And we have, as our organization, 8 scientific departments. So the workshop will be held at the regional center of Rabat, especially in biotechnology units. Our unit is composed of 9 researchers, 3 technicians, and 8 PhD students. We are working on 4 main areas. So we have laboratories for genomics and bioinformatics, where we study mandated crops at INRA. We have a laboratory for in vitro tissue culture and laboratory for plant microbe interaction, and also a laboratory specialized in plant pathology, especially in nematology. We have several greenhouses and platforms. So the aim of this workshop is to enhance participants' capacity and proficiency in ingest technologies, especially Illumina, enable participants hands-on on Illumina sequencing workflow. And of course, the objective is not only to sequence, but to handle output of sequencing and to transform the output of sequencing to meaningful information. So we'll enhance participants' capacities in terms of bioinformatics analysis of NGS data. So the workshop content is composed of theoretical sessions. We'll tackle topics related to NGS sequencing and with a focus on Illumina. And we'll give an overview of the library prep kits that we will use. Of course, tackling points related to library quality control. And finally, we will give some theoretical sessions on Linux and handling ingest data. For the practicals that will be provided, we will delve into the workflow of sequencing using the Illumina kits. So before that, we will pre-check our extracted DNA using dedicated kits and devices. We will do library prep, library quantification, and we will set up the run sequencing. So at the end of the workshop, we'll work on real data sets of NGS. So providing to the participants some hands-on NGS control quality training, genome size estimation, genome assembly. So we'll work on real data. So I think this is the content of the workshop.
Thank you for this introduction to tomorrow's workshop. Now it's turn of Professor Badawi Bouabid for introducing his workshop.
لقد الحل So hello, everyone. I will take just a few minutes to speak about the workshop that will be held starting from tomorrow in Faculty of Science, Mohammed V University here. So the workshop will be about the assessment of biodiversity through R, Python, and artificial intelligence. So we will discover how R, Python, artificial intelligence are visualizing biodiversity assessment methods and driving advancement in species conservation. So, of course, just biodiversity assessment is crucial for understanding the health and the resilience of the ecosystems, either animals, plants, or microbes, or any other species. And it provides insight for conservation and sustainable management. So what we will see the basics of R. For those who doesn't know, the software is a very versatile language, tailor-made for statistical computing and graphics. So we will specifically focus on how we can use R, some packages on R, to deal with biodiversity, to manipulate the data, and to make graphics using, for example, vegan package or ggplot visualization. We will deal too with the basics of Python language, because Python is now the most famous language for data science and artificial intelligence. So we will see some library for Python like scikit-learn and Panda and the best. So this language offers a huge flexibility and a vast library ecosystem to deal with data manipulation. And we will see that is the core language for machine learning and artificial intelligence. And at the end, we will concentrate on artificial intelligence. So we will discover the cutting-edge algorithm of artificial intelligence for biodiversity assessment. So we will explore the most known algorithm. I will concentrate on convolutional networks of artificial intelligence to cluster species and to detect species, and generally to assess biodiversity. So generally, or to conclude what we will see in the workshop, we will leverage the combined power of R, Python, and artificial intelligence to advance biodiversity assessment. So the program will be as follows. This one. So, I have to stop the other PowerPoint. It's blocked. Et la souris, il est désactivé. So here is the program. of the training. It will be done by myself, Professor Faisal Wadi of Computer Science. He will be in charge of Python language. Dr. Ishraq Hayah, he will be in charge of R and artificial intelligence and all. We will work together. The program is here. In the day one, we will speak about the morning of the day one. We will speak about the biodiversity, what is biodiversity, and we will concentrate, as geneticists, we will concentrate on genetic biodiversity and genomic biodiversity. In the afternoon, we will handle R, try to understand the basics of how we use R, how it is used. And I mean, it will be an intuitive way, because I understand that the background is very heterogeneous. So we'll be really with the basics. And second day, we will tackle the basics of Python programming. And in the afternoon, we'll speak about some algorithm of artificial intelligence. And in the third day, morning and afternoon, we'll tackle some concrete cases of analyzing biodiversity using all these tools that I spoke about, R, Python, and artificial intelligence. Before concluding, I really want to thank all the people that contribute to this workshop, but especially I have great thanks to Dr. Ishraq Hayah, because really she did a colossal amount of work. She answered hundreds and hundreds of emails. She texted in WhatsApp, she did meetings, she did all. She did more than what I did, really. So thank you very much. And I would like to applaud her. So thank you very much.
Thank you so much. So we move slide to the next workshop. Dr. Fouzia Ardouani, are you there?
Yes. Hello, everybody. Yes, I'm here present.
Okay. Perfect. I will just introduce you before giving you the...
Okay, thank you.
Okay. Okay. Dr. Fouzia Ardouani is a PhD in immunology, research director at Institut Pasteur du Maroc, Casablanca, Morocco, and head of the microbiology department. Her research interests includes infectious disease, genomics, and bioinformatics. She is conducting and managing different research projects, author and co-author of numerous publications, presented her work in numerous national and international conferences. Dr. Ardouani is also interested in bioinformatics. She is principal investigator of the H3 AbioNet project, supervising and training masters and PhD students, developing bioinformatics tools and database. She is active in H3 Africa and AfricaBP with main objective to meet the needs in genomics, data analysis, and biological mechanism exploration. So Dr. Ardouani, the screen is yours.
Okay, thank you so much. So I'll give a quick presentation about my institution, Institut Pasteur du Maroc, and a quick presentation about the workshop that that will be held in our institution. I hope that you hear me well. Do you hear me well, please? Yes. Can you hear me?
Yes.
Okay, so a quick presentation about Institut Pasteur of Morocco. As you know, Pasteur Institute of Morocco has two sites, one in Tangier, which was inaugurated in 1911 by the Spanish government, and the second one is Institut Pasteur of Casablanca, which was inaugurated in 1929 by the French government during the Protectorate. And since 1966, according to Decret Royal, Pasteur Institute of Casablanca becomes the center of serums and vaccine. And it is an autonomous public institution under the umbrella of Ministry of Health. since 1967, Pasteur Institute of Tangier, owned by APM Paris, given to the name to the Center of Serum and Vaccine. And during this year, 1967, Pasteur Institutes, the two institutes were merged to Institut Pasteur of Morocco. Pasteur Institute of Morocco is a member of the International Network of Pasteur Institutes. So what are the main missions of Institut Pasteur of Morocco? Pasteur, it's continue performing research related to the infectious diseases of animals, humans, and plants. It promotes and develops fundamental and applied research, provides a basis contribution of expertise, biological analysis, and food safety and environmental services. And Pasteur also manufacture, import, export, and distribute all vaccine serum and serums, biological products for the therapeutic and diagnosis use. It gives advices to any individual, any company, and any administration, and other regional, national, and international institutions. And Pasteur contributes to the teaching of biological disciplines related to his activities. The science departments of Institut Pasteur of Morocco are research and education department. In this department, we are holding different research activities. We are holding education and holding biosafety and biosafety practice. In the research activities, we have different projects and different laboratories: the genomic and human genetics laboratory, the microbiology, virology, and parasitology vector-borne disease laboratory, toxin and venom laboratory, oncobiology, and bioinformatics. For the education, Institut Pasteur is providing education in microbiology, but mainly bacteriology and antibiotic resistance training, in virology, in molecular biology, and in bioinformatics. To deal to these projects, Institut Pasteur is equipped by different equipment and supplies in all the laboratories, but we have a high security level Petri laboratories, which is it represents the virology reference laboratory, which is very active for all the pandemics and epidemic emergence. We have a system for strains and samples storage, and we have lots of equipment and supplies related to DNA extraction and thermocyclers and else. We have also the Genome Sequencing Center, which equipped with the high-tech technologies, mainly the Sanger sequencer, with the Illumina sequencer, the Genexus 2000, the MiSeq Illumina, the Iontoron sequencer, and different equipment for that DNA acceleration control, quality control, and sample distribution. For the biological analysis service and food safety, we have also different laboratories. Mainly for medical analysis, we have pathology laboratory, bacteriology, microbiology, biochemistry, molecular biology, cytogenetics, and genetic counseling, hematology, viral hepatitis, and else. And for the food and environmental lab analysis, we have the laboratory of microbiology and physicochemistry. In addition to all these activities, Pasteur Institute has different departments for culture media and laboratory reagents production. We have also a center for production of serum and vaccine to enable, since Pasteur Institute is a center for international vaccination, it serves for to provide advices for vaccination calendar, on therapies, treatments, international vaccination, and travel advices. So for our workshop, we will deal with the Illumina MiSeq system. The Illumina MiSeq system access-focused applications such as targeted resequencing metagenomics, small genome sequencing, targeted gene expression profiling. The MiSeq reagent enable up to 15 gigabytes of output with 25 million sequencing reads. other activities that can be provided by Illumina MiSeq. We have the MiSeq system facilitates the research with a wide range of sequencing applications. It is capable of automatic paired and read and up to 15 gigabytes per run, delivery over to 600 base of sequence data per read, and the reagent that it uses are optimized for a variety of applications, including target gene, small genome, and amplicon sequencing, and 16S metagenomics. The reagents of MiSeq allow preparing sequence libraries for small genome, PCRs, amplicons, and plasmid in less than 19 minutes, and with low DNA input requirements. The Illumina DNA preparation is fast, integrates workflow for a wide range of applications for human whole genome sequencing to applicant plasmids and microbial species. About the further application that can be handled by the MiSeq, we can deal the gene expression analysis with targeted RNA-seq, target genes panels, microRNA and small RNA analysis, DNA protein interaction analysis with ChIP-seq, and we can do also the quality control by the machine. So this workflow shows the main steps of the sequencing by Illumina MiSeq. We have the sample preparation, sequencing, data analysis, and we can also share in live the results with collaborator. The main application of Illumina platform at Institut Pasteur, this infrastructure is used for all the research projects, mainly for characterization of SARS-CoV-2 during the pandemic COVID. It's also used for microorganism characterization, bacteria and viruses. It allows handling genetic disease using target genes, sequencing for precision medicine, since we are involved in different projects with Africa and with the Africa North Africa per Medina. We are involved in this project for precision medicine implementation. So we are using the platform for this issue, for this use as well. And this machine is also used for whole genome sequencing, whole exome sequencing for the projects of breast cancer and lung cancer and other projects funded by the European Union. Now I come to the workshop and the agenda of our workshop. For our agenda, we are splitting the day to two. The morning we are, for all the 3 days, we are doing practice in the lab, wet lab practice. So we will, in the first day, we will be doing the sample preparation, DNA extraction, and quality control. And in the afternoon, we will deal with some presentation related to biodiversity, biodiversity, and we will give a lot of time to our provider for sequencing machines to give a talk. The second day, we will handle the library preparation, and we will start some tools for the genomic data analysis. Here we will choose the differential gene expression analysis of plants, and we will provide also a presentation of a test and RT-PCR, which was developed in our lab, in our institute for aviadenovirus in polyturies. In the third day, we will be sequencing and generating data in the morning, and the afternoon we will do the hands-on to analyze the generated data. So we will give the practical session for data analysis, bioinformatics data analysis. And the team involved in this workshop are myself, my colleague, Dr. Mariam Khiati, she is immuno-oncologist. Ishraq bin Amri, Dr. Ishraq bin Amri, she is bioinformatician. Dr. Abdelhamid Barakat, she is geneticist. Dr. Hisham Sharot, he is bioinformatician and biostatistician. Dr. Adil Hamouchi, who is responsible for handling the whole genome sequencing, who is a molecular biologist, and Dr. Abdulhab Benani, who is responsible for the platform of biogeomolecular. And so that's it, and thank you so much.
Thank you so much, Dr. Dwani, for this overview of the workshop. So we move to our next and last speaker, which is Dr. Eric Amombo. Are you there? Dr. Amombo? Is there any way to reach him? You're emailing him? So since we can't reach Dr. Erid Amombo, Professor Badawi will present on his behalf. The presentation is entitled Challenges and Opportunity for Expediting the Adoption of Alternative Crops in African Saline and Arid Regions from a Genomic Perspective.
Thank you. So just I will say some words about the work that will be held in EM6P Layun, Asawi. It will be held by Eric Amombo, but he is in trip, he cannot present his work, but just I will give a short overview what will be done there. So it's the work there, as is in desertic zone, is about biosaline crops or crops resistant to salinity and growth. So what will be done is that the most cultivated species in Africa, in the world generally, corn, sorghum, barley, alfalfa, are not salt resistant. So what we can cultivate in the complete desert is plants that are resistant both to salinity and growth. But improving and studying those species need to be manipulated from molecular biology point. So we need to extract DNA, to amplify it, and to sequence it. And the major scope of the training that will be done in Asawi M6P is around this. So it's what I was saying, we are looking for alternative crops to be able to live in the harsh condition in the desert. And in the Asawi Institute, they are working hardly on cultivating and improving alternative crops. I can give an example of the blue panicum. It's forage that is very resistant to salinity, and it's in very advanced stage in this institute. So I'll skip If I skip, there's nothing to say. So this just a case example. This is the work done by Eric Mumbu, and the work is published. And it's just how we can increase the sugar yield in maize. What is the genetic background underlying the sugar emitting? Well, it was a work done by Eric. Here is a summary of the work. What Eric will do also, he will speak about CRISPR-9, a technique, because he is a specialist in this technique. He will, after the molecular biology workshop, the traditional molecular biology, he will speak and explain to students how they could use this technique to modify genes, involve overexpress genes, don't express other, in the favor of agricultural trees. So that is mainly the work that will be done in Asari. Thank you.
Thank you so much for your presentation. So since we are closing this session, I ask you to remain to the stage and give the concluding remarks.
Okay, thank you. So, so we reached the end of this workshop. Thank you very much to all of you. A big thanks to the organizers, the speakers, to all the people that helped from near or far to make this workshop successful. Really, I don't want to mention names, not to forget anyone. Really, thank you to all of you. As I said, I have special thanks here in Morocco to Ishraq, and to thank God from outside that accompanied me from the beginning to the end of this workshop. It was really a huge work that takes a lot of time and effort, but at the end, we are very happy. And this workshop will be held again and again the next years. And of course, it's not the workshop that is important in itself. It has its importance, but what we are seeking is the consequence of this workshop, because as I mentioned before, we are planning and heading to make an excellent center of biodiversity here in North Africa, to have all the needed materials and support to be an excellent center for genomics of all agricultural species. Thank you very much from the deep of the heart, and have a nice day. Please, the people online, we will take photo if you can make on your camera if it's possible. Thank you.
Online participants put on their cameras, please.
Thank you for your effort.
ديري يفيكم في الورك شوب جايه مزيان صافي صافي دوك انا مقيد نورمالمون معاهم